Hello everybody, good morning. We are about to start. I'm Dr. Heidi Ekamora from the University of Atlantic Technological University. We have moved that name now. In Galway. We are going to have in our jury Dr. Cristina Ticabello from UPM, Dr. Gonzalo Machado from the University of Oviedo, and Dr. Gabriela from the University of Ghent. Okay, so Laliotti is going to be presenting a little bit of enlightenment on microfibers. Okay, so you have 15 minutes to present. When you have 2 minutes left, I'm just going to stand up in that corner. Okay, and then they have to wrap it up, and then we have 15 minutes for questions. Okay?
Perfect. Thank you. Okay, so hi, I'm Panayota, and I'm going to talk to you about how I used sediment cores in order to study the stratification of microfibers geological records deep sea. So, first of all, human activity has altered significantly the Earth's surfaces. One of the many markers of anthropogenic activity is plastics. Since the mid-20th century, we see a very high rise, exponential rise, in plastic production, which has led to the distribution of plastic debris across all major environments, from terrestrial to freshwater, from terrestrial to freshwater to marine. Even the most pristine environments are contaminated with plastics. Plastics also tend to degrade and leaving behind small fragments called the microplastics. So what exactly is a microplastic? It's a synthetic microparticle which is smaller than 5 millimeters in diameter. But let's take a step back, because not all microparticles are plastics and not just plastics are bad for the environment. We have the microparticles in general, which we can categorize by shape and by material. So we have all these different shapes. We have fragments, foams, films, fibers, lines, and pellets. In this study, we're going to be focusing just on fibers. Due to their elongated shape, and because of their shape, we can actually understand that they have an anthropogenic origin. Another type of categorization that we can do when we're talking about plastics, when we're talking about microparticles, is the different types of material. We have man-made cellulosic and synthetic particles. Synthetics are basically plastics. And both of these categories are chemically processed particles. Then we have animal-based and natural-based, which are non-processed particles, but we're going to get into this a bit later. So, let's make some connections. Deep sea. Deep sea is a massive environment that covers more than 60% of Earth's surface. It's very important because it helps with the carbon sequestration, nutrient cycling, biodiversity, but it also acts as a sink of natural and anthropogenic materials. Although plastic pollution in sea surfaces has been very well documented, we have very limited knowledge about what happens in the deep sea, especially when we're talking about sediments, and even more when we're talking about the vertical distribution of microparticles into these environments. This is because we have no standardized approaches in this field. We have a variety of sampling protocols, extraction methods, and particle identifications, which makes the whole research even harder in order to identify what we're doing. So, what is the aim of this thesis? Well, basically, by exploring the vertical distribution and the stratification of microplastics in deep sea sediments, we are trying to understand and answer just one question. Could microplastics be used as a stratigraphic marker from human activity? Meaning that could we use microplastics in order to identify a new era, which is basically signed by the human activity and the human mark. In order to do that, we have our study location. We picked the Gulf of Taranto in the south of Italy. This is a semi-enclosed basin, which means has very little water circulation, and has been signed as a site of national interest because of its high pollution, since around the bay we have a lot of industrial activity. In this location, we went to the northwest place, and we picked up this location due to the stable sediment deposition in 368 meters of depth. So, before I even arrived in the picture, in 2023, the Italian National Research Council organized an oceanographic campaign in order to understand and study the deep sea circulation, sediment exploitation mechanisms, and accumulation of anthropogenic contaminants, which basically also includes, of course, microplastics. So what did they do? They used this box corer, which penetrated the seabed, causing minimum disturbance, in order to collect the sediment, using a stainless steel line, which basically is a big tube. Inside, we have the sediment in order to be able to transfer it into the lab. On board, they sliced this big core into 48 different layers. The first 10 centimeters of the core were sliced into 0.5 centimeters of thickness, while the rest of this was sliced in 1 centimeter. And then this is where I come in the lab. So first of all, I took all my layers and I dried them in 50 degrees Celsius for 3 days in the oven, since we know that this kind of temperature is way below the limit of the melting limit of the most common polymers. Then, once dried, we did 2 density separations using zinc chloride solution, and we created 4 blanks in order to understand and control the contamination. After that, we did a visual identification and particle count that we found in our samples using the spectroscopy, basically. So, with this, we found fibers, which as you can see, and we found fragments. But we're not going to be talking about fragments because we did not identify any as plastic, and because of the shape, it's very hard to say whether they have an anthropogenic origin or not, unless they are identified chemically as plastics. So we went on to study the fibers, which is what we're going to be talking about next. In order to do this identification of material, we used microfourier transport infrared spectroscopy, MFIR basically, which basically is a machine that sends infrared light, and due to the very high origin of the material absorbs the light into different ways, creating a spectra that, according to the material, is different. So as you can see here, we have a spectra of a synthetic polymer, especially a polyester. And here, we have a man-made cellulose particle, this is viscose. The differences between these two spectra are very clear, so for us it was also very clear to identify what kind of material we had in order to do and continue our analysis. So, before we started any kind of analysis, we had to test whether our results are random or not. In order to do that, we used the Rams test, which showed a very high significance, which means that the data are rather structured than random. In addition to that, in order to be able to create the graphs and visualize even better what we call stratigraphy, because this is what we're trying to do, we did a conversion of depth into age using the sedimentation rate that was calculated on board the cruise 2 years ago, and keeping in mind that for the 0 centimeter depth, we are talking about the year of 2023. So we converted the depths into age, and then we went on into analyzing our graphs and results. First of all, we needed to assess the contamination. Do these fibers come from the lab, or do they come from the environment? To do that, we compared our blanks with all of our samples using the Wilcox test, that, as you can see, shows a very high significance and a very big difference from the blanks. Blanks basically have a very low fiber counts, and there were no synthetic particles into them, which means that we have a very low contamination during our laboratory, and all the fibers that we found, they do come from the environment. In addition to that, we measured the length and the diameter of the fibers, and we didn't show any significant results compared to the depth and the length and the diameter of the fibers. So we're not going to really talk into that because we have way more interesting things to talk about. First things first, we wanted to check the temporal trend of the microfiber concentration throughout the years. As you can see, we see a very, very high rise, starting from around the 1960s, which aligns with the exponential rise of the plastic production. But especially after the 2000s, we see an even more high rise, which also aligns with the very big need for fast fashion industry and the abundance of the clothes and the textiles that they are produced. We wanted to go a bit deeper into that, but in order to do that and understand what kind of materials and particles we have per decade, we needed to categorize all of our materials into 4 main categories, as we previously mentioned. We have synthetics, which is plastics, polyester, polyethylene, polypropylene, and polyacetone, man-made cellulosic, still playing in the chemical process category, which is cellophane and viscose, and then animal-based with wool and silk, and natural-based with cotton and linen, and stuff like that, basically. So, the first comparison we wanted to do is that we divided these all categories into two big, just to see how much are the synthetics that we find in our core. So, as you can see, before the 1960s, before the 1970s actually, there are zero plastics, which also aligns with a very high exportation rate of the plastic industry. You can see that there is a very significant, a very big increase of plastic production, and that the cellulose-based material, regardless of origin, they're constantly present throughout the entire column. So we wanted to get a bit more deep into, okay, I mean, this is cool, we have synthetics, we have cellulose, but what type of cellulose and synthetics do we actually have per decade first? So here we have our graph. We divided all the polymer categories that we found by decade. First of all, we can clearly see a pattern. Natural and man-made cellulosic fibers are constantly present through all the years. But as we mentioned before, synthetics are only there from after the 1970s. Also, something that is quite interesting is that we see a very high presence of natural-made materials before the 1920s, which could actually align with the transition in clothing textile before these years, because, I mean, people before the 1920s didn't have all the means to create all these chemical processed materials. Here, we have a heat map. It's actually very pretty and it's actually very interesting. You can see that the synthetics stop at the 20 centimeters. The 20 centimeters is basically the converted age for what we've been already talking about, 96s. And here we see a very constant trend of the plant fibers, but we have a very increased, like a very high abundance of all of them, mostly in the first layers. Only the plant fibers keep on being throughout the entire column. Animal fibers show way less abundancy, and we don't see them that often, which could actually mean that they can actually be degraded way easier than the rest of this material. So let's continue. We had the question initially, right? We wanted to check whether microplastics could be used as stratigraphic markers of the new human era. Well, the answer is actually yes. Microfibers could be indicators of human activity. We have a lot of synthetic polymers. We see a very clear pattern. It starts after the 1960s, which actually aligns with the acceleration period of plastic production, and they can actually be used as the markers of industrialization. In addition, we can see the transition in fiber composition by depth. First layers, most recent years, chemically processed, either man-made cellulosic or synthetic. Deeper you go, more natural, less unprocessed things, which actually also aligns with the historical changes in the textile production. So, one thing is for sure, we have a lot of land-based pollution, and we have a very big transportation in remote marine environments. I mean, after all, we're talking about the deep sea, and we're talking about sediments inside the deep sea, and we can still find plastic, we can still find fibers, we can still find things and particles that they originate from the humans. So we need some very comprehensive policies in order to stop, or at least diminish this phenomenon. We would filters and washing machines, we need them. We need biodegradable textiles, and we need fresh water treatments that can actually work. So in order to achieve and create a planet that has less plastic, we need some future research to understand better all of this phenomenon. We need degradation studies under deep sea conditions, because clearly, I mean, we had an entire century that we have been studying here. Clearly, they are present for such a long period. And ecotoxicological experiments in order to understand their impacts into benthic fauna are also needed in order to understand what kind of pollution and how heavy it is for the marine organism, the marine ecosystem in general. So let's wrap it up. We have a very sharp rise in fiber abundance from the mid-20th century. There is a very clear vertical structuring confirming that sediments could be used as archives of anthropogenic contamination. We see a synthetic fiber trend that aligns with the global plastic production, and microfibers, which is the final conclusion, could actually be used as stratigraphic markers of the human activity. Thank you. I'm sorry, I cannot hear you very well.
Which is the degradation time of all the fibers into this
Well, so far throughout the study, we didn't really see a very big degradation, especially when we were talking about plastics. They did not degrade. The shape was the same.
But if you know from the literature or from other studies, if you know that it is very significant.
Well, regarding plastics, we're talking about centuries that they take to degrade. But when we're talking about man-made cellulosic fibers, like viscose stuff, because normally for solids, they're not considered as contaminants. There are no studies that they actually show how fast or how long they degrade. We did not find much literature about this. But for the plastics, we're talking about centuries.
Yeah, so first of all, congratulations on the presentation. I really enjoyed it. Really nice way, quite energetic. So, putting our attention to the content. So, congrats about that. So, I have some questions. How did you choose where to take the samples? Because what I would like to know your opinion about the repeatability inside, because as you know, there could be some places with higher position. Which one, if you were to take like an average, because it's clear there's no certification, you nicely show us that. But if, let's say, if I have to see or to perform, to continue this study, which area should I go? Should I take different samples from different areas and do an average? If I don't know if this is a position area or not, what's your opinion about?
Well, first of all, to answer your question, this is part of a bigger project. So I only work with this location, but during the cruise, they picked up locations from all over around the area, basically. So honestly, if it would be me, in order to have a better view and understand better, I would pick a location that is very close to Taranto, because we have a lot of industrial activity there, and there are a lot of rivers and runoffs. So if I were to study and see a very clear pattern about how much is the contamination into the water, I would pick up here. But I mean, that's the goal I have to work with. But for future studies, I would go closer to the shore.
Yeah, and related the question was related to the currents and how different currents might distribute plastic, even though the origin could be very close.
Oh.
How different current might generate some deposition areas even far away from that area.
There were not massive currents. Like there was. This is why we have a very stable sediment deposition. There are no currents. The base is enclosed, so you don't see a lot of movement from the plastic, from the materials and the seabed. It's pretty chill. So it doesn't really.
And then the other thing is, I work with some people that some microplastic. I know it's really time consuming. It's really weird. So, do you think, what's your opinion about automatization of the process? If it's automatization, automatic somehow, do you think it's feasible? What's your opinion about that?
Well, actually, there are a lot of machines, even FTIR machines, that they are automatized. And I was in a workshop a week ago actually talking about that. So, well, it fastens a lot and speeds up the process, but it doesn't have the same accuracy. It's different because we did it one by one, checking the video, the image, and I mean, it took ages to do it. But you're actually there and you do see it. The machine sometimes crashes, you leave it, it doesn't understand some particles, the lengths are not well measured, so yeah, it really works and it's a very good solution in order to speed up the process and have a lot of, actually be able to test a lot of samples. But in my opinion, it's not as accurate as doing it manually. Also, there are a lot of different techniques. We have MFDR, we have ramen. Ramen is better sometimes. It depends what you're actually searching for. Like ramen, you can actually see and measure microfibers and microplastics in general, that they are smaller than 10 micrometers. But also, it doesn't give this wide range, and it's not as accurate, the spectra, as it is with the FTIR.
Okay, thank you very much for the presentation. Really a lot of work, so congratulations. Did you analyze all the 40 years?
Yes, I counted 1,200 fibers and 1,468 fragments, but the fragments were not plastic.
Yeah. So I'll go a bit more technical. So you analyze them at the microscope, but usually you consider that a little bit higher than human error. So was there any validation methods applied?
Well,
to make sure that what you counted was actually plastic and wasn't something else that looked like plastic but it wasn't.
well, you mean during the
Identification.
with the microscope? Well, at the beginning that was very hard because I got the sample and everything looked like it's man-made or plastic. But reading a lot of papers, photos about how they actually look as how is a fiber, how is a fragment that you should see. After a while, the process became easier. And before I started my experiment, I did do like 2 weeks of testing into different particles in order to be able to validate that, okay, what I see now, this is not just a stone or something. But there were a lot of random stuff. I picked everything. Like I didn't leave a single fiber behind. So then I just tested all of them into the FTIR in order to see, oh, well, this is plastic. Ah, this is not plastic. This is nothing, and it's just in my head. So, yeah, there is a very big thing of human error in the microscope.
If you think you left something behind that you thought that's something not plastic.
No, I picked up everything. Literally.
Good. And then something else. So we have a lot of organic matter in your sediment samples that interferes with the FTIR. So we're wondering during the density separation process with this flora, did you also do something else with the organic matter or was not?
Oh, yes, of course. We do.
Sorry.
Yeah, no. I did not explain it to that part. We did the two density separations, and then we filtered our samples twice. So all the sediment was left away, and we had like how do we call it? Like cotton, like the filters. Thank you. Yeah, we had the filters where only the particles were left after our filtration. Then this, we took it to the stereo microscope, we extracted everything, and we put it in a very small petri, like a glass, with just water, in order to let it dry, in order to use the FTIR machine. All the sediment was removed before.
And then I have a question about the blanks.
Yes.
Did you have blanks only in the lab, or did you also have blanks on board? I know it's less common, but did you have blanks on board?
No, I was not there for the cruise, so they just gave me the sediments and the blanks were all made in the lab, which I do understand that it's way more accurate if you make your blanks on board because there are exactly the same conditions, but it didn't happen and I just created them in the lab. Because also, I mean, they were all processed in the lab, it's not like they processed anything beforehand, so this is where we wanted to see what kind of contamination we get.
Yeah. It's just that very recent papers say that the airspray from the sea that goes on board actually has a lot of microplastic contamination.
It's true.
And, but it is really not used yet.
Yeah.
But we should improve it, so that's why I was wondering. But anyway,
Yeah.
okay. More about the area, because it is considered deep sea because it's 398, but do you think, what's the difference with really deep basins? So let's say 1,000 meters, because in a lot of papers that consider salmon as safe for microplastics, they're actually talking about 1,000 meters of deep basins, like in the open ocean, for example. And here we have a coastal basin, so you would expect more sedimentation, but do you think it's comparable? Do you think you can apply the same rationale to deeper basins, or is that not? How does it compare?
I think you can actually apply it. You just need heavier and bigger machines, and the extraction will be 1,000 times harder, but I guess you can. And regarding all the other processes, it's the same way, the same lab, the same extraction methods that you would use. It's way harder. The conditions are definitely not the same. We have way less oxygen. Maybe we have higher degradation or lower degradation. But unless you try it, it's... For currents, well, I guess it depends on the area. I mean, when you go so deep, you have to check the currents and understand. The sedimentation range will always be different.
suspension.
Oh, the
of the
suspension.
surface, because we found most plastic on the surface, which is
Yes.
wrong. But then
Oh, you mean that all this could go into a deeper layer, like the deep?
yeah, depending on the characteristics of the area, then there's more suspension and then they transport.
Oh, yes. Yeah, the deeper you go, I guess, the less transportation it will be. Like it's a seabed, it's a floor, they just stay there. So possibly you can find even more plastic if we go deeper in the ocean.
Can I ask some questions?
Yes.
Do you want to ask one?
I would like, what is the first year that you into the lab?
Here, 80, 70s.
And the fiber at that time was
Well, they were mostly plant, like just plant, not man-made, no plastics.
is it possible that the fibers are not completely this time?
Yes, actually it is, because you have deep sea conditions. It's completely different than the water column. The oxygen is very low. We have a completely different environment. So probably, maybe, I mean, from papers that I read, this is not exactly clear. But because of the conditions, it's possible that they did not migrate. And since we had a very, very low contamination, they have to come from the environment. It's actually impressive how from the 1970s you can actually find still cotton in your sediments.
Do you think it's possible that, I know that obviously we've had a rise in mass production and the less movement is but do you think that the rise in fibers all of a sudden is only due to the fact that there is a rise in production of plastics, or because only now we're paying attention to that?
Oh, I definitely believe it's because we're not paying attention to that. I mean, when you have samples from the 1970s, that means that it was there and nobody cared to check it before. Of course, the industrialization has become way bigger due to like socioeconomic reasons. We buy more clothes, we spend more, we buy way more like fast fashion clothes. We use a lot of laundry machines. So yes, there are a lot more textiles and particles that they fall into the sea eventually, but it's been there for over a century. So I just think we did not care and did not pay attention to it before.
Just one last question, because you're mentioning bioplastics as a sort of substitution for the solution. Do you want to expand a little bit more about what you think about bioplastics?
Well, I don't think it's a solution, and I don't really think that it actually works, but it's definitely better than all the kind of textiles that we have at the moment. So, honestly, I haven't really given it a thought into what type of alternatives we could have. Probably, I mean, you see way less contamination when we're talking about animal fibers. So, this could be a solution, in my opinion. Like, they tend not to spread that much and they stay longer. But biofibers and alternative like recycling material, I personally, I think that yeah, it is a kind of a solution, but it doesn't really solve the problem. It just creates a new one that we're going to face in 20 years.
Thank you. Okay, we'll be ready for the next. Okay, so we're going to be hearing now from Natalie Mayer about plastic pollution as well. Are you ready?
Thank you. All right. Hi, everyone. I'm Anna. I will be presenting my thesis today titled Understanding Coastal and Offshore Plastic Pollution Patterns at a Continental Scale. To start, I wanted to show you this figure. It's by Geyer et al., and it shows the historical in blue and the projected in red dotted line global annual production over the years. We can see that ever since 1950, the production has increased exponentially, and it is also projected to increase up to 1.1 million metric tons in 2050 if we continue this business as usual scenario. This is an issue because higher plastic production means that there will be higher plastic waste and more waste leaking into the natural environment, and especially the aquatic environment. We know that plastic enters the ocean through multiple different pathways, such as urban river runoff, stormwater drains, direct littering, the shipping and maritime industry. And we also know that about 80% of plastic that we find offshore is land-based, so from the coast. That's why it is very important to understand this very dynamic relationship between coastal and offshore plastic pollution. And I used Australia as a case study to investigate this. It is important to investigate this because we know that plastic in the natural environment affects and harms wildlife, affects human health, and also sectors like the tourism industry. All right, now to quickly talk about my aims and hypotheses. So my first aim was to quantify plastic distribution in coastal and offshore environments around Australia. And I hypothesized that with increasing distance from cities, coastal and offshore plastic densities would decrease. This comes from the main hypothesis that we have higher waste when we have more humans living in an area. My second aim was to describe the relationship between this coastal and offshore environments regarding plastic. And I hypothesized that plastic hold in hotspots, so areas where we find lower plastic densities and areas where we find higher plastic densities on the coast, match those offshore. And this comes from the main hypothesis that just like river plumes, the density by the river mouth of nutrients, for example, is very high and then dissipates out into the ocean, and this can similarly be seen for plastic inputs into the ocean. Okay, I want to talk about my 3 data sets that I used for this study. The first one is the National Coastal Survey. They conducted around 170 surveys around Australia, and with multiple transects per survey. They recorded metadata on every survey and transect, and then recorded all the plastic particles that were found, and categorized them into different categories, such as plastic, and then types for plastic. Those were, for example, fishing line, hard or soft plastic, and so on. And then I came into play and wanted to visualize and analyze this data. I, first of all, mapped all of my data that I had, so in this case, coastal plastic densities, similar to this map. In this map, you can only see the locations of the surveys that were conducted. And then I selected 10 urban coastal cities to conduct my analysis. What I did was I classified transect sites that were within 100 kilometers of a city as inside the city, and all of the transect sites that were between 100 and 200 kilometers of a city as outside of the city. I then calculated mean plastic densities and standard errors for both of these categories and each city, and then calculated the ratio between inside and outside to understand where we had higher densities, either inside or outside. Now let's move on to the second data set I used, which is offshore sea surface trawls. So now we're looking at offshore data that was actually observed. They conducted manta and Newson trawls with 3 to 5 consecutive tows per station, and then counted, measured, and categorized the plastics per tow, and then calculated the densities based on GPS coordinates. Again, then I came in, as I did for the coastal data, mapped this data and selected the same 10 cities to conduct my analysis. I wanted to understand the relationship between the distance to the city and the plastic density we found. So I calculated the distance for every trawl location to its nearest city, and then conducted a GAM, a generalized additive mix model, to understand this relationship. I used a Tweety distribution with a log-length function, as this accounts for the multiple zeros in the data sets, and then used cruise station as a random effect because we have this nested experimental design. The predictor variable was distance to city, and I used it in a smooth and a linear term to test which model worked best, and then used the AIC and in coordination with the significance of the predictor variable to understand which model was best. And the third data set I used was actually a model, so an offshore plastic particle tracking model. This is a model output sourced by Clark et al. 2023, and they calculated the arithmetic mean out of 3 different previously conducted models on plastic particle density estimates. They all have different assumptions, and that's why they combined all of them together. In the end, they had this oceanic grid that estimated plastic densities around the world, and I cropped this to my desired area, which was around Australia. Then I came in again and used the same 10 urban coastal cities. I classified all of the grids that were close, so the 3 grids that were closest to a city as close, and all of the grids that were directly adjacent to those close grids as far. And then again, similar for the coastal data, I calculated mean plastic density, standard errors, and the ratio of close to far. Now let's get into the results that I obtained. I'm going to show you the results per data set, just so we can understand the singular ones, and then show you the whole map as a whole. Now we're looking at the observed coastal plastic patterns that we saw, and we can see a few hotspots, especially around Perth, on the western coast of Australia, then southern Australia, and also on the west coast of Tasmania. Looking at the 2 classifications I made, inside and outside of the cities, we can see that Perth in purple has the highest mean density per category, so in both inside and outside, and the lowest densities are described for Darwin and Exmouth for the inside category, and Darwin and Karratha for the outside category. This is interesting because in this map, we see Perth had very high levels of plastic pollution, and then Karratha and Exmouth are pretty close together, and Darwin is pretty far up here, and they have very low populations. So, and also we can see like very low densities around them, so that does make a lot of sense. And then let's look at the ratio that I calculated. We can see I added a 1 ratio line here, so anything that is above the ratio means that we have higher densities inside a city, and anything that is below the ratio means that we have higher densities outside of the city. In this case, we can see 6 out of the 10 cities have higher plastic densities within them instead of compared to outside of them. And this supports my hypothesis that with increasing distance from cities, in this case, coastal densities decrease. Okay, let's move on to the offshore plastic patterns that they observed. These are the trawl data that I looked at, and again, we can see some hotspots, especially in the north of the East Coast. This is around the Great Barrier Reef region, then the New South Wales coast. We have a few hotspots around the west coast of Tasmania, and also some hotspots around Karratha. Now looking at the GAM that I conducted, I chose the best fit model, which had 76% of deviance explained, and this was for the linear term for my predictor variable. We see, in this case, sadly we don't see, but the slope is at 8, and this line is already at 30,000, so it's very difficult to see, but I can tell you that we have a weak but significant relationship in this case, meaning that with an increasing distance to a city, we have lower plastic densities. And this, in this case, again, supports my first hypothesis that I made, in this case for offshore plastics. And now let's look at the modeled data that I used. Again, we can see some hotspots. In this case, in the southern Indian Ocean, we can see a little bit of a microplastic reservoir. Then in the Great Australian Bight, around Adelaide, and in the Bass Strait of Australia. Looking at the categories, we see that Adelaide in pink has a very high mean plastic density, the highest out of all of the cities for both of the classifications. And in this case, Karratha shares the lowest mean density for both of the classifications. Looking at the ratio that I calculated, in this case, half of the cities show higher plastic densities inside of, or close to them in this case, so close in their offshore waters compared to far offshore waters. In this case, I can't really support my hypothesis. All right, let's compare these two ratios because my second hypothesis was that the cold and hot spots would overlap. In this case, half of the cold and hot spots overlap. For example, for Brisbane, we see higher densities inside the city. on the coast, and also for Brisbane, we see higher densities for close offshore waters. So they would overlap, but there's other cities where this is not the case. Sadly, this doesn't support my hypothesis. All right, let's combine all of the data that I used in my thesis in one plot. We can see the 3 different data sets, and also, again, the cities are marked in green with their population. I want to make 3 main assumptions here. So, the first one is we see higher coastal concentrations compared to the sea surface. So for both the observed data and the model data, we have lower densities compared to the coastal data. This is the first observation I made. The second observation I made was that the west coast and a little bit of the southern coast look very different in their plastic distribution compared to the east coast. So what we see in the west coast is we have this high reservoir of microplastics here, and we know that the leeward current travels in this direction. Now, I've read a few papers that state that a lot of plastic gets caught in the heel of Western Australia, so in this area. So this explains the very high densities we found here. And this also explains why we have higher densities around the Great Australian Bight modeled and the trawl data around the Western Australian coast, or Tasmanian coast, being a little bit higher than the rest. The Zeehan current is the southerly extension of the Lewin current and also supports this hypothesis. So we can see that, especially also on the rest of the coast of Western Australia, we have very scattered distribution of plastic, and here around Perth, it's very high and very aggregated. So I state that the western coast is more governed by the oceanic processes and also a little bit, obviously, the higher population that we find around Perth. And the east coast of Australia shows a very much higher population in general and higher tourism for this area. So we see that in this case, the coastal plastic distribution is very evenly distributed, and that kind of supports this idea that with even population numbers, we have evenly distributed plastic. But also we have higher density of trawl plastic density in the Great Barrier Reef region, which is a high tourist region, which then makes also a lot of sense. So yeah, these are the 3 main takes I have for my data. Does this really support my hypothesis that plastic cold and hotspots on the coast match those offshore? I feel like in some case they do, and in some case they don't. There are a lot of dynamics that are in play here, especially the touristic region and the population, the different currents we see. Therefore, it's very difficult to pinpoint why these patterns exist, but I feel like I've made a good assumption here. All right, in conclusion, just quickly, I wanted to show you my 2 A's that I had. So I did quantify plastic debris distribution around Australia and the coast and offshore and describe the relationship both of them have. I couldn't really support or dispute my hypothesis. I feel like there's a lot of other work that can be made to understand plastic distribution patterns. But yeah, I feel like I did make a good introduction to that. And yeah, thank you for listening.
Congrats for your presentation. That is very, very nice. And which are your hypotheses to support your presentation?
So my main, so what I found to support my hypothesis is the question.
You show up that there is no strict correlation with the population density, for example, and the plastic that you found cities, right?
For the, well, let's go back. So for the offshore I did find a significant relationship, but it was weak, but there was a significant relationship between the distance to city and the density of plastic. So that, I would say, does support my first hypothesis.
Yes, but if you show the last,
This,
the last slide, please. Yes, this
Yeah.
one. Yes, I can see the population
yes,
is the density, because density is higher in the south,
yes.
but for plastic, the hotspots of plastic are in the north. Right?
For the offshore density, yes. That's, I hypothesize that that is due to the touristic region of the Great Barrier Reef, and there's a lot of tourism coming in, a lot of people going out on boats and trying to see the nature, and therefore there's a lot of plastic produced.
So you think that this is related to both tourism?
Yes, and also there's a city up here, Cairns, which I know shows very low population, but there will be a lot of people flying into Cairns. Also, Townsville is a little bit further south. So, yes, I think it is connected to boat tourism, but it could, yeah, I mean, probably mainly connected to boat tourism because that's how they go out and explore the Great Barrier Reef. Yeah.
Yeah, so first of all, congrats on
Thank you.
great job. So 170 places, it's really
Yeah.
something that is huge.
Yeah.
So in that sense, well, and I guess this is part of a big project and soon, so well done on that. And can you comment to us about certain temporality or heterogeneity due to time? How, if it's somehow, at least for some of the sites, was tested, the difference along
Yeah.
the year or something?
I did not test that at all. All of the data, so the coastal data was collected between 2011 and 2013, for example. So this is like more than 10 years ago. And obviously there's probably temporal differences throughout the year, especially with rainfall, where more plastic gets put into rivers and then into the ocean. But I did not test for that in my thesis.
Do you think that it might have a strong
Yes,
effect?
I think
or-
so. I think it does have a strong effect. I think especially in rainy seasons, we would probably see way more plastic coming into the ocean than in dry seasons. Just because a lot of plastic gets put into the ocean through rivers. Yeah.
Another thing that I was thinking, what do you think about, not only because here, and it's a lot of effort that you get so awesome, amazing, but how do you think that you can get extra information if you analyze at least, not for 170,
Yeah.
but at least for some critical, the types of plastic and the match between the coastal and the offshore, if
Yeah.
that could inform and also type, if there's a match with the type of microsoft from the city comparison with the coastal, and what do you think that, how can that inform to you, not only just the number, but also the type of
Yeah.
plastic?
I think in some ways, it probably can inform, the type can inform on what kind of activities are happening in specific regions. In this case, for the coastal data, it was not just microplastic, but also bigger, mainly bigger plastic pieces. And then for the offshore, it's mainly microplastic just because of the methodologies that are used. It's very difficult in the offshore to collect bigger plastic pieces, which I would definitely say is a limitation of the study. But I feel like with a few pieces, I mean, I feel like it's always difficult to say because you can differentiate, for example, between land-based and ship-based plastic, for example, but a lot of plastic that is used on a boat is also used at home. So it's really difficult to say how it got introduced into the ocean and to validate that. So I don't know, in some ways, maybe if you find like a plastic shovel that could have been used on the beach, then yes, that would inform that. But if you find a plastic bottle in the ocean, you don't know where it came from and how it got there.
Then, how would you integrate somehow your model or for testing also because you have your elements, I was just trying to check the main carrots there, so it's not the ideal dynamics of
Yeah.
the dot, it's quite well known. How can you integrate that into your analysis? Because you might expect that there's certain region is well known, which could be acting as a source,
Yes.
some sort of accumulation area. So how can you integrate that into your model to correct for something, some areas where you might expect some certain accumulation due to current, some areas that maybe there's a lot of microplastics, but that are just
Due to the currents
travel.
travel.
So
Yeah.
how,
So I think I would actually start with the data that I used. So for the model data, for example, I calculated the arithmetic mean out of 3 different models that all assume very different ways of how plastic gets introduced into the ocean. So some of them the assumption was that plastic was just introduced everywhere around the whole ocean, which isn't really the case. Some of them stated that they had plastic particles introduced in major cities and around major shipping routes, which would make sense. And I feel like in that case, the currents could come in, and you could add like another layer of assumption that the currents would travel in this specific direction and take the plastics that route. But this would have to be like a fully new model to make. I used an existing model. Another thing I wanted to say about the model was that it was obviously in like a global model that I used, and I cropped it to a specific size of Australia. So this would not be as how do you say, like specific, as specific to like the Australian region, and is more broad. So I think that is another thing I would change to try and make it like specific to the Australian continent, and then add the currents maybe. Yeah, thank you.
Thank you. It's an insane amount of data that
Yeah.
you analyze. It's really all different data sets, which it's already hard in microplastics. We have one data set.
Yeah.
So with different data set, I can imagine that it's been a huge amount of work. So congratulations, please, and give you the presentation. So I agree also that it's really hard to connect onshore and offshore, because even if you have polymer type, you don't know if actually that polymer came from Japan.
Yeah.
So
Yeah.
I understand. So let's start from the data set. So because they were different data sets, did you use the same size range of plastic, or because the net was 320 years?
Yeah, depending on which study was used. So the trial data was from different cruise ships, and different nets were
Do you know
used.
the range of those?
Yeah, so 333 to 350.
Yeah, so that's our sort of limit for that, and
Yeah.
then surveys, it's probably mostly
Yeah,
macro plastic.
so they are
And then the
different.
model, what was
The model is also microplastic.
micro?
Yes.
So lower than 5.
Yeah.
Okay. Yeah. So that's also
Yes, that is a limitation of the study. Yeah.
That's what's right. But what I was thinking, in addition and comparability, okay, we always have that in literature.
Yeah.
But do you think that also influenced then the distribution of the plastic? So when you serve, it's also influenced by the size in the data set.
Yes, I would say yes. I mean, it is very different how a bottle reacts compared to a particle in the ocean. It's like that, obviously, the spatial component is a very big one that I didn't really account for in the study. Yes, I would say yes.
And then another thing, so you have high concentration of plastic closer to highly populated zones, which is usually what one might assume just because of price and stuff like that, right? But have you thought of the opposite? Because we always think of plastic going into the ocean.
Yeah.
But how about the plastic
Coming onto the ocean, onto the shore. Yes, I have thought of that because there's also hypotheses that, or studies that show that this connection between population and density of plastic does not exist. Like both has been found in studies. So, sorry, the question was if onshore plastic. So, yes, I've read a few studies also by, that were supervised by my supervisor that looked at a student was at the beach and marked all the plastic that she found one day, and then she came back and checked what had arrived and what had left. And it was, I think it was very specific to the type of plastic that they found. So yeah, if it was a bottle and there was a lot of wind, then the bottle was gone probably the next day. But for bigger things, they stayed. And I think she also found a bunch of things that came onshore. So it really depends on the currents, I think, the weather conditions, like there's so many dynamics that are in play. But yeah, I did think about that, and I also discussed that in my discussion in the manuscript because it's an important part of the whole dynamic. Yeah.
If there was a follow-up on the seasonality also.
Yeah.
If storms, for example, if there were and then the observations.
Yeah.
I understand.
Yeah.
It's a nice yeah.
Thank you.
Can I just ask, I must have missed in the beginning the origin of all the data? I understand that you collected some of it.
No, I didn't collect any data. They were all data sets that already existed that you can find like open access online. So the model data is a source from Clark et al. 2023, which they have like calculated the means of the different three models. And then the trawl data was a global data set from Erickson et al. I'm not really sure about the dates. And I had chosen the area around Australia, and it was mainly data from 2011 to 2014. And then the national survey is from CSIRO, the Commonwealth Industrial Research Organization in Australia. Yeah. Which is the National Science Agency of Australia, and they had conducted these like 170 surveys around, and that was from 2011 to 2013. So it's all a very specific range. And the model was also like a still picture from 2014. So it's kind of a similar like data range, I would say, or yearly range, which was important to me. Yeah.
Okay, so Janet Catherine is going to talk to us also about microplastics. Whenever you're ready.
Yes. Good morning, everyone. My name is Lena Schaffel, and I'll be talking also about microplastic, more specifically the exposure risk of microplastic contaminants on filter feeding megafauna in the Azores. I was supervised by Dr. Christopher Pham and Dr. Jorge Fontes from the Instituto do Mar and Oceanos in Porta in the Azores, as well as Dr. Javier Hernandez Borges from the University La Laguna in Tenerife. So, as we all know, plastic pollution is a big problem. It's a growing problem in our oceans, and a lot of plastic that enters the ocean accumulates in so-called oceanic gyres. So these are plastic accumulation zones. We have 5 of those in our oceans, and one of those is the North Atlantic subtropical gyre, which you can see here on the map. And the Azores, my study area, lays on the northernmost boundary of this gyre. So obviously the Azores are also highly exposed to plastic pollution. But the Azores are also a migratory stop for many filter-feeding megafauna, such as the sickle-fin devil rays, the Mobula tarapacana, which we can see here in the video. And they are quite vulnerable due to their feeding strategy, the filter feeding, to microplastic pollution, as well as the particle prey size overlap, so the prey that they feed on with the microplastic particle size range. We know the Azores are a biodiversity hotspot, but I'm wondering, are they also a plastic hotspot? And are these animals at high risk of ingestion and therefore contamination from microplastics? To study this, I have 3 aims. First off, we wanted to quantify the microplastic concentration in the surface, but also the subsurface at a specific aggregation area of these filter feeders, such as the sickle fin devil rays I showed in the video. Secondly, we wanted to characterize those microplastics for the physical properties, which is the size, the color, and the shape, as well as the chemical properties, such as the polymer composition. And last but not least, we wanted to evaluate the exposure of plastic-related chemicals on these filter feeders, such as the Mombula tarapacana. So the study area are the Azores archipelago. We can see here on the map, they consist of 9 islands near the mid-Atlantic ridge. And as I said, they are a biodiversity hotspot. And I want to highlight here we have the central group of the islands and Fayal and Pico. And then one very special aggregation area of these filter feeders is Princess Alice Bank. It's a seamount around 60 nautical miles offshore, southwest of the central islands. Here again we have Fayal and Pico, and here the red is Princess Alice, that seamount. So what we did last year in the summer, between July and August, we collected samples. We were trawling there, as well as one control area, which is the purple dots here. And in total, we collected 43, we conducted 43 transects on the seamount. And how we did that is followed the transect, the contour line here, and tow over the summit, and always crossing the summit with every transect to cover as much area as possible. And how we did that, we applied different sampling techniques. So obviously we have the manta trawl, which is a very typical sampling technique, which is shown here. So that's being towed on the surface. And then we also have a bongo net, which we deployed in the subsurface at 3-meter depth. And both of these nets have a mesh size of 200 micrometers, so 0.2 millimeters. And we towed them both for 15 minutes at 2 to 3 knots next or behind the boat. Since we are sampling for larger than 200 microns, we also collected some water samples, some grab samples with just 5-liter water bottles to also see if we find particles that are smaller than 200 microns. And just to get a better idea of how this towing works, I have a video. I wasn't sure that's audio, but we have the manta trawl here on the surface, the bongo net here, deployed at 3-meter depth with propellers running, so we are going and towing over the summit right now. Okay, and thirdly, we also collected biopsies to check for the chemical exposure to the sickle fin devil rays. I have a video showing Bruno Macena, one of the researchers in Horta, collecting a biopsy. So he goes diving with a spare gun, selects an individual, and then he shoots and collects a small biopsy of 1 centimeter from the pectoral fin. And then we saw whale shark, which was really exciting. This method is non-lethal. It's low invasive, fast and efficient because they do it free diving. All right, let's have a look at some of my results. So first, so my first aim was to quantify microplastic at this aggregation area. So on the surface, so with the manta trawl, on the surface we found particles in all toes. In total, we collected 543 particles with the surface toes, and I calculated the concentration to 1.06 items per cubic meter at Princess Alice seamount. For the grab samples, since it was way less volume, so that was 5 liters per bottle, we had a high concentration of 820 items per cubic meter. And then for the subsurface of the bongo net, that was 3 meters below, we had a statistical significantly lower concentration of 0.01 items per cubic meter. And then now here looking at the graph, this is showing all the different campaign dates where we sampled last year. And we see the concentration here on the y-axis there was no statistical significance between the, I have to explain, the red is the control site and the blue is the Princess Alice, but there was no significant difference. So, but it's just showing the temporal variation. And for example, on one day, we had a really, really high concentration in one of the flows. We had over 100 particles that we found, which is quite interesting. Okay, moving on to some other categories. So the shape. On the left, we have the subsurface, and on the right, we have the surface. In the subsurface, we mostly found fibers, so the fibers are in red. In the surface, we mostly found fragments, which makes a lot of sense because fragments float and fibers sink. Also, the graph samples, which are not shown here in the graph, we found mostly fibers, which could be due to that the net doesn't really contain those fibers because of the water pressure that those fibers get pushed through the net, even though they're larger than 200 micrometers. Then also looking at the shape together with the color. So the predominant colors we found are white, which also includes no color and transparent, as well as black, dark, and blue. And we can see that there was not a big variation between the control site here on the left and Princess Alice on the right, but obviously some of the shape differences that we've seen here are also shown there. So now moving on to the size. So you can see a high peak here at just above 1 millimeter. So that's 1 millimeter and above is the size category of zooplankton mostly, which also is a big part of the prey that the filter feeders feed on. So we have this overlap of the size and the prey, which can of course lead to accidental ingestion. And overall also on so the blue is the subsurface and the red also showing high percentage of larger pieces, so all the visible ones above 1 millimeter. And now looking at the polymer composition, so we have on the surface, we mostly found, so in orange is the polyethylene, so one of the polymers. We mostly found those in the surface, and in the subsurface here in blue, we only found cellulosic fibers. Makes sense again, on the surface, polyethylene are mostly fragments that float, and the fibers sink, so they are in the subsurface. So yeah, let's have a look at a bit of the why is this? The mean concentration that we found at Princess Alice is above average comparing to different key aggregation areas of filter feeders. So there hasn't been any study done in this area looking at the concentration, so I compared to different regions, Mexico, Indonesia, Galapagos Islands. In Indonesia, this was the paper I found the highest concentration of, which was up to 0.9 items per cubic meter, and my concentration at Princess Alice was above 1 item per cubic meter. So it is similar, but the concentration we found was still a little bit higher. We found no significant difference between Princess Alice and the control site. We did see a few temporal variabilities that could be influenced by local hydrodynamic features that affect plastic transport and accumulation around the seamount or in other areas. And as I've shown in the map in the beginning, the Azores are positioned on the North Atlantic subtropical gyre, which support this regional vulnerability to plastics, especially floating plastic debris. And we also saw a vertical stratification, so between the surface and the subsurface, all the polyethylene mainly is quite positively buoyant, so we found a lot of that on the surface, but not on the subsurface. Now looking at the biopsies we collected, so out of 25 biopsies, in 9 of them, we detected phthalate esters, which are plastic additives that are added to plastic during the manufacturing to make them more flexible and less likely to break. And we detected the most common compounds in 9 biopsies of those, and DEHP in 5 individuals. And these ranges that are shown here in the table, so up to 36.2 nanogram per gram, are within similar range compared to other ecotoxicological assessment in also other filter feeding species, but at low concentration. So overall, it's quite low concentration, also compared to other studies. Some of the ecological implications. So as I've mentioned, this was the first ever study done in the Azores at the study area and the first ever detection of phthalate esters in the Mobula tarapacana species from the Azores. So I can say that there is environmental exposure to these animals from plastic-derived chemicals. I've mentioned the plastic particle size overlap with the prey, which can lead to accidental ingestion. And due to the ingestion, obviously, we have physical but also chemical risks, which can lead to long-term health concerns such as oxidative stress and endocrine disruptions. Some of my limitations are that the sampling was done in the summer months between July and August last year. So seasonal trends are unaddressed, but also the devil rays only aggregate in the summer months. So that is the only time, at least for the biopsies, to collect. Of course, if we continue the manta trawling and so on, before or after that is still possible, and that could give us more insight into the abundance of plastic in that area, also in different seasons. One thing I want to highlight is that during the sample processing, we did not do a digestion, which is crucial and which we should have done. But we didn't do it from the beginning onwards, and we didn't want to add it halfway through to change the procedure. So there is, since I did it all manually, all the identification and also the microarchaea, it's possible that I missed particles. And as we saw before, there was a lot of visible particles which are easily identified and less invisible, so smaller than 1 millimeter. In terms of future directions, of course, it's important to continue the study, to do long-term monitoring, not maybe at this seamount only, but also at different seamounts. So there's many seamounts around the Azores, so it would be interesting to also look at seamounts, other seamounts where these species and other bitter-feeding species aggregate. It would be good to continue the development of this non-lethal sampling method, and also to maybe widen to other megafauna species. We've seen that there's many whale sharks to found also at this seamount, but also other seamounts. So it would be interesting to maybe also collect biopsies from them and check for chemical exposure. And then just to conclude, so microplastic pollution is pervasive at Princess Alice, but not exclusively, since we've also seen concentrations at the control area and most likely at other areas if we sample there. So I cannot say it's a super dangerous area for these animals for plastic pollution, but we are aware of this plastic pollution problem. I've mentioned the particle price size overlap, the exposure to both physical and chemical pollutants, and the need for standardized long-term monitoring for risk assessment and conservation, and the non-lethal biopsy sampling, that it's been proven to be effective and scalable and should be continued. Thank you very much. Supervisors and all the people in the field and in the lab that helped me collect this data. Greatly appreciate it.
Christina, would you like to ask a question?
About the future directions.
Yes.
Do you think it could be useful to examine the gut contents of the zooplankton?
Yes. So we also, when we were trawling, of course, it's not the only thing we collect is microplastic. When we are trawling on the surface or the subsurface, we are collecting everything. So we also got a lot of phytoplankton and especially zooplankton. And the idea was to also check them for, so kind of do the same procedure we did for the biopsies, but also for the plankton. But in the end, there was not enough time. But the sample is still in the freezer, so this could still be done, and it would be really interesting to also, because of course, that would be like the trophic transfer. If we already find chemical traces of plastics within the plankton and other species feed on the plankton, it can have even worse effects. So yeah, that was an idea.
It's nice because we could integrate.
Yes, I'm actually going back to the Azores next week. Yeah. Yes. Thank you.
Yeah, so congratulations to them. I really like the presentation. Also, I like your presentation that you really show the limitations and think
Yeah.
it's nice to discuss it. We should never hide our reputation in our studies.
Yeah.
You just
Yeah.
put it there, and I think make your study somehow by showing.
Thank
So congrats to that. Also congrats on the amount of work and for doing that presentation. I really enjoyed it. So thank you very much. So one in terms of predictability, or did you have some plans?
Yes. So question came up before. So on the boat, after we took the nets out of the water, we opened a air contamination control filter, one for each toe. And those obviously I also checked back in the lab for particles, and I think in total I found 5 or 6 particles in all of the air contaminants. So that was really low.
I was surprised to see the controls in the graphs that you show us. You were not yet, no?
No, but they were so little. Like in most there was nothing, and then 1 fiber there and like 2 fibers there, which can obviously come from the air or maybe our clothes.
Yeah,
but it's important to check.
I will include just to
show.
have clear impact as having, it's like almost,
Yeah.
it was sealed, right?
Yeah.
So,
Yeah.
but I will show.
Yeah, and we had the air control on the boat and the lab, so doing the lab processing, the filtering and so on. I also had one.
But also to show both level of potential error that in your case was almost almost nothing.
And then, so a lot of people call these blanks, but for me it was like the air contamination control filter, because we also did blanks for the water, since it was already filtered water that I was using, but we still had to check that, but we didn't find any contamination.
Okay, that's great. So I also one thing that I also like when you talk about plastic, so that you put it into context. I really like that. Like, okay, in that particular vision, this is the expected number per cubic meter or whatever. So I think that gives some context to say, okay, how much higher or lower is that? I really appreciate that. And particularly to that topic, because one of your hypotheses is that due to the area that you are studying, these currents traditionally. Do you know, or are you planning to test within that area, but in different positions, if that might change or not in concentration?
Around the seamount or in the old islands of the Azores?
Just to put it in the context within that particular region.
I think that's really important. So now we really only sampled at one seamount, and then basically on our boat right back to the island, we stopped halfway to the control site. But obviously the control site, we are not sure so much what is going on and whether there's something else going on, why there would be higher concentrations. So it would be really good to have different locations. I know there's some data, I think from 2022, where they collected south of Pico, so one of the central islands, where there's also a whale aggregation area. So another area of interest, but I think in general, checking other seamounts or just other areas without special features to really be able to compare, I think would be really important. Actually, what we're going to do this year is take a boat and sample all around the island of Fayal, a bit more coastal and a bit more offshore, so a little bit like Anna's, to really see how also that differs. So we will do lots of manta trawling. Yes. Thank you.
Great.
Thank you. Very interesting study. Benthic ecologist, so it's always nice to see some water sometimes.
Yes, deep blue water.
So first of all, actually, I just see it, so you did the digestion of the plankton, and it's also very hard because it has to be mathematical at this point.
Yeah.
But how do you think it's actually affected your samples?
So in the beginning, we had very clear samples. There was not a lot of organic matter. That's why we didn't just start with the digestion right away, but it should be the common procedure and should be done either way, which,
We have a protocol, so I understand.
Yeah, okay, anyways, so in some samples that we collected, I had to do 2 filters because I was filtering and there was so much organic matter, so the whole, like if you look at the filter, it's basically like brownish green. So if I put that, and it's almost like a layer. So if I put that under the stereo microscope, of course I can see maybe some of the white fibers, but the ones that are more brown, I don't see. So of course that could have changed a lot of the numbers I use now. So.
And did you wash them? The zooplankton, because I know sometimes when you do enzymatic digestion, you just wash the animal with microplastics
It was very small organic matter. Like we didn't have any, I don't know, are you referring to within the water sample?
Yeah, so when you collect the flow
Yeah.
and you have your zooplankton soup,
Yeah.
and then you have microplastics flowing in the
Yeah.
water, but sometimes if it really can get attached to the skin of the
Yeah, so we actually found more like blue dragons. I don't know if you know these, like, yeah, we found some of them. Yeah, we killed them. Anyways, we removed them before I did the filtration. So any larger animals, let's say, we removed, I washed it and removed it.
already that's
Something, that's something, yeah.
And so you said you had some lines in and what do you think we should do with those lines? Because we did find some fibers, they weren't that many, but we found some fibers.
Yeah.
So,
for this case, we didn't actually do anything. Of course, I mentioned it also in my manuscript that we found 5, 6 fibers, but I know like the blank is correlated to a specific trough. So what we could do is see if there's overlap and then maybe subtract the fiber.
What do you think about subtracting that? It's a debate in my
Yeah. Like I have discussed with my supervisor that it's okay to just say like we have found a few contamination fibers and like that's enough. But I've also heard other people say, so I cannot really form my opinion because I've heard like both sides, and for me I understand both. For this case, we decided to go not with the subtraction because I think it can be other factors like or changing other factors. So if I would have to choose, I would do it how we did it. But I also understand why you maybe want to subtract, but I guess then it depends on the amount you find in the
Yes, and what we subtract the bottom, the size,
Yeah.
the type.
Yeah.
So in the literature, because I have no clue, of course, about your species, did they already found plastic in the tissues and
No, this was the
so
first
this is
episode.
like the pilots that
Yeah.
actually for the species.
Yeah. Like they've done, like in other, in the manta ray, in different areas, they've done biopsies collection or other filter feeding species. In the fin whale, in the Mediterranean, rare species where we collect the biopsies, but never on this species in this
Do
area.
they know the impacts on this?
So the researchers, such as Bruno from the video, they are very knowledgeable, very skilled, and this is really a super small like stress event that they experience. And I have these Vertebrate experts sitting at the back, so please correct me if any of this information is wrong. She did a on the species also yesterday. But yeah, it's a small little, and they swim off, and there has been no like behavioral or other effects from this.
Yeah, sorry, maybe I wasn't clear.
Okay.
No, I meant in the other, in their studies of the impact of microplastics on the species, or similar, yeah.
Like in the tissue or?
Yeah, well, because
Like
we studied the presence of microplastics.
Yes,
in animals,
yes.
But does it actually happen?
Yeah,
Because
so
sometimes some animals just
that's for the ingestion and like the physical harm. Of course, you can like collect in the stomach, but since I looked also at the chemical effects, because these chemicals like these phthalates can transfer through the gut wall into the bloodstream and reach all tissue. That's why we were sampling muscle tissue to see whether we find any concentrations there. And just as in humans, it can have, as I say here, oxidative stress and endocrine disruption, so hormonal changes, which can lead to behavioral changes. But this area is super understudied. Like this is really where we're stuck kind of in this research, and we don't really know, like, okay, it's affecting them, how much, to what degree, like, are we seeing behavioral changes, is it causing cell damage, like all of these things. It's really not out there yet.
Yeah. And so, since you found really low concentration of these chemicals, though, in your opinion, is it actually going to be? I know you don't have a lot of data, but just in your opinion, based on literature and everything, do you think it's actually going to get a impact, or it's something that the animals will just be resilient enough to go through?
Of course, we don't know. Since we sampled in July and August, and these animals come in June, because they only aggregate in the summer. Of course, it could be only low concentration because they only arrived recently, and maybe if we could sample in September when they're still around, maybe we would find higher concentration. But of course, we cannot make the link. I cannot say because there's this much abundance in the waters in the Azores and there's this much concentration in the animal, like we cannot link it. It could be from where they came from in February that they were feeding on macroplastic in a different area where they were before, and that's why it has accumulated in the body. So of course, combining this information with maybe tracking, like where these species come from, sampling the area where they were before, it could give us more insight. But of course, we cannot say the concentration. Yeah, but just detecting something already means they are exposed to microplastics at a low concentration. But thank you so much. Yes.
So you found a certain number of persistent organic pollutants that you were saying there from the biopsy.
Yes.
Okay, so one of them is making you believe that they have ingested plastics. Is that?
Say,
One of these contaminants is making you believe that they have ingested plastics.
so these compounds are in use during, like they are added as plastic additives, so-called active esters, in the plastic production. And what can happen is that either during, like when the plastic piece, like when a fragment has been ingested, these toxins from the plastic can leach off, either within the stomach and then leach into the tissue or already in the water. And these compounds can also adhere to to natural fibers or other particles in the water. That's why we didn't exclude the cellulosic fibers. And therefore come into the body through the ingestion. But of course, there's also ideas for that it's not only ingestion, but maybe the intake or other ways of, let's say, ingestion, active or passive, that it could enter the body. I don't know.
Yeah, no, you did answer. It's just, I mean, they are persistent organic pollutants. And even though they are used in the production of plastics, they're persistent in the environment.
Yeah. So they can also be found in the water, of course. Yeah.
So it's, obviously, we know that they are exposed to microplastics.
Yeah, so it doesn't always have to originate from the plastic itself, but it can be from the water and then get to the.
Just be cautious with the statement, like this is evidence that they are,
Okay.
exposed to microplastics.
Okay. To microplastic contaminants. Maybe it's better to break
Yeah, because
yes.
it's everywhere.
Yeah.
It's persistent.
Yeah.
You don't think about that.
Okay. Okay. Thank you.
Thanks very much. All the speakers and presentations.
Is that true? Yeah, he needs it. We need it also. Yeah, but that'll be funny.
It's not the hottest room.
It's not the worst one. And it's still morning, so it's still not that bad. But I insist for the deliberation, you want to fund and each one of us. Most of the viruses, but there's an operator. I think you can go online. Yeah, that's what I got. Okay. So welcome everyone to this last session. As usual, we have introduced us this morning. We have Christina from Neopelagic, Jesper from Ghent University, Heidi from TPU, and myself, I'll be chairing the meeting from UBO. As usual, you all have 15 minutes to present. When you get to 13 minutes, I will try to wave and get your, I will wave and try to get your attention so that you know it's time to wrap up. And if you see me standing up, to that you're mostly over time and we can finish this as soon as possible.
Okay.
And then we'll have a 15-minute discussion with the members. Okay. Whenever you are ready.
Okay. Well, good morning, everyone. My name is Antonio Ignacio Garcia, and I'm going to be presenting my ongoing master thesis project based on the taxonomic and functional analysis of the newly termed Neopelagic community associated with the floating and stranded anthropogenic marine debris in the Madeiran coast. But before I start delving into the impact of the marine debris into the ocean ecosystems, we first need to understand how some species have naturally adapted to survive in this vast, faraway coastline ecosystem. So, historically, it has been assumed that a species with larvae planktonic dispersal has shown a bigger capacity of distribution, let's say, in contrast with the species that didn't have this mechanism. So, like species that were more like direct development. And several studies through the decades have shown how several studies have shown how several studies have shown how this assumption was incorrect, since there's been I cannot. It has been shown how is it different, basically, from the
Take a breath.
several studies have shown how this assumption was wrong. This assumption was wrong, since some species have different distributions, which cannot be assessed with the typical larvae planktonic distribution. And this cannot be explained since ocean rafting, as you can see in this graph, was discovered, and it shows how the organisms without this type of mechanism can spread farther abroad. And what is an ocean rafting, let's say? Well, an ocean rafting is basically a biological dispersal mechanism in which organisms such as algae, plants, vertebrates are able to be transported around the marine environment, with what we call natural occurring rafts. What are these examples? Well, basically, we have here some examples of vegetative fragments, such as coconuts, tree trunks, or sargassum rafting, very famous in the North Atlantic. A seaweed known to hold a huge amount of a huge community assemblage with both coastal and pelagic species, such as Geniella tuberculata, and branches as Neopelagic. And then, in contrast to this rafting, which are of organic nature, they're facing biodegradation, or like megafauna predation. They are known to be very short-lasting rafts. In contrast, we have others like pumice or epibionts, from volcanic sources, or epibionts, which are able to last longer in the journey around the raft. All of this in conjunction with highly energetic events such as tsunamis, cyclones, hurricanes, has shown how some items, as you can see here, this would be the rafting, some items are able to cross natural biographic barriers that would explain very clearly how we can see continental coastal species in very far oceanic islands. So, the anthropogenic debris, well, we've known that for this past decade, a lot of solid waste has been dumped into the ocean, up to 4.3 million tons of debris, and it's known that the 70% of it is at least plastic, highly buoyant, and extremely durable plastic. So these act as a permanent island accumulated in the gyres and able to hold these coastal species. To study this anthropogenic marine debris, it is divided in floating marine debris, beach marine debris, and seafloor marine debris. So as we know, floating MD, permanent refugia for these coastal species, but it's important to note that as well as it happens with sargassum, for example, different species of sargassum hold different type of community, different type of substrate, different type of the journey the item went through the ocean, not only through the ocean, but as well through the water column. It would shape the community that we are going to end up analyzing, or that it will end up reaching the coast. So we can divide the floating debris into two type of items. So like we have the ocean traveler, plastic items able to endure these long ocean journeys, and then locally sourced debris, which has a higher level of surface to volume ratio, which basically means that they are very thin, and as long as they start to be fold, let's say, as the biofilm weights, they sink very quickly. It can either partially sink or completely sink, depending on the water column. We would say have one community or other type of community. So the seafloor, if they end up sinking or not, or partially sinking, they will end up stranding in the beaches, as well as the floating can directly beach without sinking into the beaches. This is when the coastal colonization occurs. When they approach the coast is when all of this process of invasiveness occur. So when they strand, they can turn to be newly stranded debris, which just arrive. And these newly stranded debris can either refloat back into the sea and keep on traveling around the ocean, or it can be stranded at the end of the beach And there it would face desiccation of the soft tissue organism, predation of the organism, and therefore a loss of taxa information. So historically, the pelagic area has been conquered by the Neostone. Neostone species was the majority of the taxa in the ocean, with scarce natural rafts around the ocean. But now we are facing this situation in which we have this accumulation of trash in the gyres with permanent coastal species that were not known to survive in the open ocean, interacting with these Neostone species would have done a total community shift. As we can see in here by Haram et al. 2021, we can see species as well that we can see in Sargasso, such as Geliella tuberculata, Planes mytilus, and so on. But taxa is important, but more important is the functionality of the community. So, like with this plot that Gibson et al. In 2006 she studied the functional diversity of the fauna found in the natural rafts. So you can see here feeding behaviors, reproductivity behaviors, as well as development. Therefore, this would be a very valuable information to know what is going to be the functional impact in the oceans. How to understand how they manage to create self-sustaining coastal communities on the high seas. Not only on terms of the feeding behavior, but also how they reproduce, because it's very important to know how they reproduce in these gyres. Since, for example, it can act as what we call stepping stone process, in which larvae organisms can be a steady source of propagos. So now it's not a coast-to-coast invasion, but like a colonization of different debris all around the gyre. So amplifying even more this invasiveness effect. So yeah, plastic not only is a physical and a chemical pollutant, but can act as well as an invasive vector, which is an additive to all the problems we already have. In this plot by Ramalosa et al. In 2025, he showed a model with theoretical particles were plotted in the Gulf gyre, representing the debris, the floating debris, and we can see how Madeira is in the hotspot of all the arrivals. So it's in high risk of arrival of invasive species. They show how after doing, even though 95% of the floating debris pass by through the northeast current, in the biogeographic analysis from the debris they collect in Madeira, less than 4% here, just less than 4% of the invasive species were from that area, which will explain how the item also needs to be in steady waters. Because since this is a high speed current, there's no possibility for it to be colonized. So most of the organisms that they found come from either the Mediterranean, from the Canary currents, or from the white Caribbean area. But as well, one of the species they found was called Isognomum bicolor, and it was the proof that there can be, as this species was never found in Madeira, they can be it can be assessed, tracing back the history of this taxa into, as it comes from the Caribbean, it can tell all this stepping stone process coming from Azores, then Portugal, and Spain. There is, apart from being a primary Nis vector, it can also act as a secondary Nis vector. As I mentioned before, we have the locally sourced debris that is very localized, doesn't spread around the ocean. So this can enter or get out of the ports, which we know there's a lot of invasive species there, and it can spread as well either from the seafloor or from floating locally sourced debris all around the local coastal area. So current issues. There's a huge lack of monitoring, even though we have harbor Nis monitoring in Madeira, there's no rafting monitoring for Nis early detection. If there is any kind of sampling, it's usually biased towards the beach marine debris, not the floating debris, and usually only taking into account either just trash or just the sessile fauna. Never into the mobile fauna detection. And when there is a mobile fauna analysis in floating, for example, usually they don't take into account in the methodology, but the mobile fauna jump I chased a lot of amphipods during this study. But yeah, so what would be our introduction? Well, our objective and hypothesis. The objectives: analyze the taxonomic and functional composition of all the Neopelagic community we found in the Madeira archipelago, in both floating and beach. And then understand, well, two of our hypotheses would be that the beach marine debris doesn't reliably represent this Neopelagic community. So we will be losing information on what is arriving to the island, and as well, if there is any pattern of co-occurrence between beached and floating debris. These materials and methods. The sample was carried out in Madeira archipelago. 5 beaches were sampled in Porto Santo and in Madeira, and 14 objects, as you can see here, floating objects were collected during the experiment. For the beach sampling, very easy. Government, it was carried out in collaboration with government samplings, monitoring samplings of the beaches, as well as our research team. It was opportunistic, so it was we took only the items that present biofouling on it, just walking along the shoreline, and every item was stored in individual plastic bags to avoid cross-contamination in between items, as well as taxonomic loss, taxonomic information loss. And it was divided in both not newly stranded, so the debris that was in supra-littoral area, that's been there for long, and what we know that is newly stranded, at least for the last 24 or 12 hours, because it appeared, as you can see in this picture, in the last tide line. For the floating marine debris, as well, opportunities in plastic bag to avoid cross-contamination and loss of taxa. GPS was recorded, and it was 2 approaches. In one, we went with the Observatorio Oceanographico de Madeira with the boat searching for debris, and as well, I developed this flyer in order to outreach the skippers and whale watching companies, so like with our contact and the methodology process, so if they could help us in to have more chance of finding these floating debris. Both type of debris was processed in the same way, so it was washed in shift, inspected, and then taxonomically identified. The mobile fauna was sorted and counted as well, and then very important, we took high-resolution pictures of the different species. I will later on explain why. The same, once the item was washed, we also carry out light lab item inspection. We would categorize the item according to the OSPAR guideline. We took pictures if there was any code, any labeling that could tell us the origin of that object. It was inspected, and the different thing here was that we took as well in situ photography in order to give a context to the stereomicroscope photography of the different species. As well, we carry out buoyancy tests. So like if we could assess if the item comes from the seafloor or it came to the beach by floating, by drifting. All these pictures were used to carry out a PowerPoint, which was sent to different experts in order to get reliable species identification. And there was that when identifiable for whatever reason, lack of characters or any reason, was sent to Lisbon, to a Mare Research Institute, in order to carry out DNA with this organism. This would be my data set, abundant presence and absence per object, and all the species on the left side. So samples, basically, every single item would be a replicate. So as you can see here, the sample distribution. From the floating debris, we collect 14 replicates. From the newly stranded replicates, 20 replicates. And not newly stranded, 58 replicates. And as you can see, these are all the beaches we sampled. They were quite uneven according to the locations of beaches. But as it is an opportunistic experiment, there's no problem because we are going to analyze our location. It's going to be the whole archipelago, and what we are going to compare our factor is going to be the types of debris. Oh, sorry. Sorry. And basically, yeah, the experimental design was very simple. So, like, as I say, comparison the different type of debris and the analysis that we were going to carry out is still on the way. Univariant for species richness, mobile fauna abundance, using primer. and the multivariant analysis with Permanova to analyze the community structure in general. So thanks you all. That's all.
Thank you.
And reference.
As in football, I added a little bit of time to let you catch your breath, but
Thanks.
somewhere like even with that,
Yeah.
it was a minute and 30 seconds over time.
Okay.
With added time.
Okay.
Thank you very much. I'm going to turn over to the members of the jury. I don't know who wants to go first. Lady? Yeah.
Antonio, thanks very much for your presentation. Well done. I think that kind of research is extremely important and very, very interesting because it's something that we do have to do with not just, kind of stage that it's happening. So I just have a couple of questions. First one is why did you only look at mobile fauna?
Well, we only look at mobile fauna mostly because of time. This is a taxonomic experiment. Taxonomic identification takes long, and we didn't have the tools to do the coverage of the sessile fauna in the time. So we decided to go for mobile. I mean, we're taking like sessile and mobile, but the abundance is only going to be analyzed in the mobile fauna. But in terms of taxonomic identification, just the present absence is sessile and mobile. Yeah, both.
And so far, like how many species do you have?
So far, we have 140 that we think, but identify for sure till genus level around 80. Around 80 species. It's been really good, the identification. We are collaborating as well with the University of Seville, and they are experts in vertebrates, the Malacostraga. So they're helping us to identify that is the most complicated at the end, taxon.
Did you mention anything about molecular analysis?
Yeah, DNA. We're going to do DNA with things that are maybe very degraded in the beach marine debris, because of desiccation or predation. So we're using that to send it to the lab. It's a lab from Madeira, it's that I am, and they are collaborating as well. So they will be doing the identification.
So is this a project that already exists and you just maintain, or is this something that is going to move further or
This project is Patricia Ramalosa, the one I mentioned that had the plot. He's been getting these debris for like since 2014, but he only analyzed sessile fauna, and the mobile fauna, as he's not an expert as well, he's a technician, so he cannot reach farther than family, let's say. So how my project will be different is the focusing on the mobile fauna, and like the methodology we use in which we collect the debris per item. With the bag, we avoid this, because at the end when you collect the floating debris, as soon as you take it out of the water, mobile fauna starts to get out. So you're losing abundance and taxonomy information. So that's the different approach to this study.
And are you collecting any information about the debris that they were in?
Yeah.
Are you collecting any information about the debris that they were in?
The debris, the item? Yeah. We're categorizing the items, but we're measuring size, length, and yeah, it's like OSPAR, I don't know, it's OSPAR guideline. It's like a guideline the European Committee did for standardizing, yeah. So we're following that process.
So just one last question. So, okay, you got these results, but what do they actually mean? Like, how are they going to help?
How are they going to help? Well, basically, so far we have found, so it's two approaches. The one is, the main objective is assessing the difference between the beach sampling and the floating debris, the beach debris and floating debris, and to see which are the taxa that are lost. That's the co-occurrence point. Like, which are the taxa that differentiate one to another when they get stranded. That's why we also sample newly stranded and not newly stranded, to see kind of a gradient in within the floating when they just arrive and when it's older. And then another objective that we found actually at Tanaidatia, Teuxo, that has for the first time been recorded in Madeira. So in that sense, we're also trying to reach the maximum level in order to be able to identify if we can invasive species, new invasive species that could have arrived.
You want to find invasive species that are...
And that's as well, but it's kind of an aside because that's very opportunistic at the end. You can or you cannot.
Okay, but what can you do with that information?
With that information,
What does that mean for us?
it's a stakeholding at the end. Like there is my laboratory is really involved in the politics of the island. As I mentioned before, there is already programs into the monitoring of the harbors. So they have like plates in the harbor place. So it would be a way of pushing the politics into, and also involving with the flyer as well, involving like the local community for like the skippers and the whale watching. Also, we're thinking of giving them kind of a reward in the sense of like they can show that they are helping us, because at the end for them it's marketing as well. It's like ecological. So like it also involves these companies, this monitoring. At the end, basically is to assess a monitoring because it's in sirens, it's in a hotspot of all the arrival of the dead. That would be the end. A little part of it, but
About this last aspect, I was curious about the science aspect and how the community participated in your project. Do you have difficulties in involving engaging people?
So far, we knew from, as I mentioned, Patricio, the technician's been doing this for long, so he had a list of people that already collaborate with the lab. And all the whale watching companies we went through, because here is a big industry, they were super happy to help, and there was really no issue in approaching them. And it was showing also for the clients that they were helping to, and in general in Madeira, the community is super involved into this beach sampling, and there's a lot of talks as well in cafes about science. It's really nice community, and they engage really nice.
About the types of insects,
About them?
the kind of elements.
Mm-hmm.
Did you notice some relationship with the kind of beverage and the community that wasn't associated to material or the shape or size?
As I say, it's a very stochastic. Process, because as I say, it can beach and then refloat again, and then it can sink, and then it can refloat again. So it's very complicated to track back the story of the item, let's say. But we with traces of knowing that sometimes taxa appear. Usually, but this I couldn't do the stats still. Like this is gonna be for the future. But for what I see from my observation, it's true that you can see how debris, for example, that are deep in the beach, that maybe they come in winter. We have a lot of storms. They come after the storms, and they usually come in patches. Like the debris is not like scars. They usually aggregate as well with sargassum. So you can kind of, from my perspective, I repeat, see some common features in between the organisms from the same beach, let's say. And regardless the type, we just took plastic. We didn't took wood. We were just focused on the plastic. But yeah, there's been studies that show this importance of the microstructure of the, as I say, like the structure is super important for the larval settlement. So it definitely have an impact on the community as well. Mm-hmm. Yeah, and also the first arrivals. If the first one arrives, it's like pioneers. Briar swans usually enhance the biodiversity. The lepas, you see, but lepas and barnacles don't like each other. They don't really. They don't get along. I don't know why. They don't appear never together.
Thank you. We have time for two. I know you have some more, but
thank you. Thank you for the presentation. Really interesting stuff. subject, showing that dispersion can occur in these new mechanisms. Okay, so I was wondering, time is an important factor for these new drafting substrates. Would you measure time on the beach? You measure when it arrives on the beach, you have like two distinctions. You arrived and it was already done for a long time. I think the key in this is to understand how long did it travel? How long did these substrates travel in the ocean? And how long was the community able, and from there was it able to grow? How would you test this? How would you test? How long has it traveled?
How to frame that? There is a lot of, we weren't able to catch one, but there is this program, it's from NASA, that they release GPS located structures, and you can trace them. It's called Ocean Travelers, and you can trace them where they are. So you can see if some are passing. Actually, I think Patricio got one, but they when they pass close, you can collect it, and you have all the way the item did. But it's true that it's really complicated. It's really complicated to know all these.
One more question. So we have these new rafts. They can disperse very long because they can float very long. Is this positive or negative for biological communities?
Okay.
Why positive, why negative, why both?
That's quite ethical because there is this ocean cleaning NGO that is cleaning all the Pacific garbage patch, and it's quite an ethical question. Like, it's already a settled community because you can see in many articles, you can see that there is this interaction between limestone and the coastal species. So I mean, at the end, I think the impact of the plastic as physical and chemical pollutant, not only as invasive, is also a good point to.
But more focused on dispersion. So we have higher connectivity, larger dispersion distances for organisms. Is this good or bad?
Well, that's like it's enhanced the invasive capacity of many species. Yet Madeira, like Macaronesia, is full of now Caribbean. So at the end, islands are very fragile ecosystem, full of endemisms. And you get a lot of competition with the native community. So if I would have to say, I would say like it's not very good to have this steady source of propagals, because it's not like from the Caribbean to Azores or to this constant spread already. So yeah.
Thank you very much, Antonio. Whenever you are ready, you can go ahead and I'll start.
Okay. Hello everyone. Today I'm going to present to you my thesis. So it's assessing environmental plastic exposure in two vertebrate groups in the southern North Sea, more specific microplastics in seal species and macrolitter in bird nest. I was able to do my thesis at Flanders Marine Institute in Ostend. So a little background introduction. We all know that plastic pollution is quite a big issue these days, with a lot of it not being recycled, ending up in the environment, often ocean-based, but also land-based. And if it's land-based, it often runs through rivers, through estuaries into the ocean. Often estuaries are seen as hotspots, since because of the tidal cycles and everything, it ends up there, and there's actually an increase in amount. Since plastics are lightweight, durable, and durable. They persist in the environment and they can also fragment over time, giving several biological implications. For example, entanglement injuries or digestion problems, which is physical exposure, but you also have chemical exposure, often from pollutants or additives in association with polymers or plastics. Another one, which is a bit more subtle, is food web transfer. So when a low trophic species eats something, it can go up to the food web, having a plastic exposure at higher levels as well, which brings us to vertebrates, which are often seen as bioindicators of plastic pollution in the environment, in several studies. So this brings us to the species of interest. So on one side, you have the Canadian goose, which is the ground nesting species, with a quite early breeding season. And then on the other side, you have the grey seal and the common seal, also known as the harbour seal, which are the two species present in the southern North Sea. Going on to the research gap and the study aims. So plastic pollution is actually studied quite a lot these days, but there is still limited data on plastic and wildlife interaction in especially estuarine and coastal zones. But also there are a few studies who actually investigate and link the local litter levels. So why was this study done? It was to link environmental plastic levels to wildlife exposure, to evaluate vertebrate as indicators of plastic pollution, and to support monitoring and conservation efforts. So how was this done? It was done through the analysis of seal species to look for microplastics in them, and on the other hand, it was bird nest photographic imagery in combination with environmental litter from an ongoing project in the area. For clarification, all the slides about the seal part will be with the blue circle, and all the slides about the bird part will be with the green circle. So going on to the seal part, the sample collection was done in collaboration with 3 sanctuaries. So two of them were along the coast, which actually take seal pups in when they are sick and then release them back. So we asked them to collect the first species that they leave to show what they ate inside, in the wild. And there was also a collaboration with Azorean Sea Park, which has 2 captive seals, and these seal species were used to test methodology, but also to check if they also would ingest plastics. The circles on the map are the spots where the seals were rescued, so where they were found. During sample collection, there was also on-site collection, so to check if there's contamination during the sampling process, and also per batch of processed samples, there was a procedural blank to test for contamination during the process. Then the samples were dried and weighted, followed by a double digestion with H2H2 and KOH, followed by a Nile red staining to bring out the colorization of the plastic particles, leaving them on a filter with all the remaining debris. These filters were tracked under a fluorescence microscope, and an image series under the 3 filters was made. So 1 filter of UV, green, and blue. This image series was used in a random forest model which is based on the fluorescent coloration of the different particles to see if it's actually plastic or organic material and which type of polymer it would be. Then additionally, there was also a verification done with microsphere, which actually looks at the spectra of the polymers and combines it with a library to see what is comparable and what could be this type of plastic. To test the method verification, so we used the captive seal species to test the methodology, because this methodology was originally developed for water samples and not this kind of matrix. So some samples were spiked with plastics, which were known, and then these were checked later on. Going on to the bird nest analysis. So here you can see the area where it was checked. Was in the Azorean in Newport in Belgium. This area was actually chosen because there is an ongoing treasure project which tries to reduce the outflow of plastics from rivers to the North Sea. So there is a lot of data available on this region. So what was done, the nests were spotted, they were captured with a picture, and it was also already investigated which bird species, which was always a Canadian goose in our case, which types were present and so on. The analysis was done following the treasure protocols, so all the found debris was categorized in J-list categories and then were scored with possibility of or likelihood of possible sourcing. Here it is to visualize where the other sample transects took place, so one at the beach and then one more inland and then also one during the bird nest sampling. Going on to the results, so for the seal species we always had a filter with all the remaining debris and always the photo series of the different filters. For the recovery test there was an average recovery of 100%, but of course it was a super small sample size and it was mainly to develop the methodology and to get to know everything around it. But on the pictures you can clearly see that the colorization of the different particles is quite clear and can be analyzed. Continuing with the samples from the wild and the captive seals, so a total of 19 samples were processed, including the blanks and the procedural, the blanks and the on-site blanks, with a result of 259 prospective plastics. All of these plastics were included in the distribution. No, sorry, not all of them. First, the plastics inside the blank samples were checked, seen if they were matching with the ones from the actual samples to account and exclude them for further analysis because they're possibly from, or most likely from, contamination. Then there was a color distribution made for the fibers because for the particles it's not super clear which color they originally were. So the fiber was mainly blue and black. Then going on to the results of the model and the FITIR analysis. So I want to say first that it was randomly selected, so some of them were analyzed by the model alone, or by both of them, or by either of them. So for the detection model, it showed that 22% of the found prospective plastics were actually plastics, mainly PP and PE. For the FITIR outcome, you have also 20% mainly PP, but also a lot of minerals. For example, sand was one of them. Going on to continuing with the results. So for the FITIR analysis, it's often used as a verification method, which allowed us to extrapolate the data of the actual analyzed samples to all the prospective plastics, which resulted in a mean of 2.30 plastics per gram of dry weight. So when you look at the captive in comparison with the wild seals, you can see that there is a slightly higher mean concentration, but this was not statistically significant. Same when you look at the wild seals only and compare between the species, you can see that the common seal could have a higher concentration, but also not statistically significant. Continuing with the bird nest analysis, so 4 bird nests were found. In 3 of them there was plastic seen, categorized into 4 categories. So fragments, dolly ropes, and one other that I forgot now. But also with three colors, so orange, white, and blue. Here you see a table of overview with the litter categories also in the nearby areas, for the top litter categories mainly food wrappers, plastic strings, and fishing nets. And then for the top probable sources, it was mainly because of households, fisheries, and aquaculture. Continuing with the discussion, so a little overview of the results. There were plastic detected in 10 out of 11 wild seal samples and 2 out of 2 captive seal samples. There were more particles found than fibers, and the main found polymer types were polyethylene, polypropylene, and PET with a concentration of 2.30. In previous studies, for example, on water samples or sediment samples, there was already done a lot of investigations on plastic polymers, which were actually often the same ones that were found in these seal samples. Also, particles were often more seen and different colors were also kind of similar. But then also some prey species were already investigated in this area, also showing comparable results in terms of found plastics, which shows both exposure pathway of direct and indirect interaction. So this shows a trophic transfer which could be supported by the overlap in prey polymers and other characteristics. Also, you need to keep in mind that also the captive seals were exposed because they also had plastics inside their seal fishes, which is not super clear if it would be from their food alone or it could also be from their habitat inside the sea park. And then if you look, for example, to a study in Antarctica, the concentration in this study was higher, most likely because there is more anthropogenic interaction. Continuing with the evaluation of methods, I think it was a very good idea to actually combine the model and the FTIR and the visual inspection because it gave a combination of everything and both had their own strengths and limitations. For example, the FTIR is quite detailed, but it is quite time-consuming as well, and it's sensitive to background matrix, which makes it hard for these kind of samples because, as you can see on the picture, there's a lot of debris remaining, which makes it difficult to see what's underneath the layers. Then for the model, it's quick and scalable, but it's not actually developed for fibers and dark particles, which is also a quite high limitation. Then if you look at the results of both of them, you can see that 21% was sea plastic by the model and 22% by the opposite, but by the FTIR, which is actually quite similar. Continuing with the bird nest, so 3 out of 4 bird nests there was actually plastic found, which actually also matched with the plastic and litter which was found in the area. So it is actually a good indicator of what's happening in the environment. One thing that I want to say is that in a lot of recent studies, birds actually show selectivity in what plastics they incorporate inside their nest. So, for example, they like a spring, or they see that it's a good structure for their nest. They actually chose it, and it's not a good view of what's happening in the environment because they actively choose what they will use. Also, checking the methodology that was used. I think photo-based monitoring is a quite good idea because you have an unlimited data frame, which is always being there, and it was quite easy to take the pictures, and it's also not super invasive for the birds. And then there are, of course, a lot of future recommendations. It was, for example, combining the photography with checking, combining the photography with actually checking the nest and seeing what's inside, because of the structure of a nest. It's of course 3D structure, so it's better to also check inside, and of course use do it several times, several years for several species as well. So to put both discussions together, they both showed a good complementary indicator, but please keep in mind that the birds could be selectively choosing. And it's a low disturbance and practical fieldwork, and it also integrates long-term and exposure view. So it's showing the complete ecosystem because they are on higher trophic levels. So for the microplastics in the seal species, they were analyzed in all, but polymers reflected environmental and prey species plastics, both showing direct transfer but also indirect transfer. For the nest, they contain filaments and fragments, matching local litter, showing clear evidence of habitat contamination and their environment. I think both are still quite good as bioindicators, but maybe it's often also just better to just investigate how they interact and not see them as actual bioindicators. And of course, more research is needed in this field. I think that was it. Thank you. And of course, thank you to my supervisors and everyone who guided me along the way.
Thank you, Lydia. Nearly perfect use of your allotted time, so congratulations on that. Maybe we start with Christina.
About the results of the nests, maybe how could you exploit the results to reservoir nest evaluation to limit, for example, the liquid dumping in the river?
So I didn't mention it, but the actual sample collection was during a cleanup. So it's every year that they actually clean this nature reserve, and so that's why we were allowed inside as well, because normally you cannot enter. So So there are actually already a lot of people who are investigated into this cleanup, and they already came to us to ask like, oh, what are you doing? Why are you allowed to take pictures? Because you were not really allowed to take pictures. And I don't know, I felt like they were already investigated into the problem, and then there were also a lot of people talking to us like, yeah, we're like from the, I don't know if it's from the government, but people from the city, which were like, ah, so your project is on this, and I don't know. I felt like they were already very interested, but of course, I think if I would go back to them now and say like, okay, these were my results, even if it's not a lot, I think they will really like to, yeah, I don't know, take it to a bigger public and, yeah. Yes, yes. Yeah. Mm-hmm. Yeah. It is very sad to see that they do this cleanup every year, and every year there is more. So it's quite sad. Yeah.
Yes, thank you for the presentation. Do you like this very clear?
Thank you.
A question, maybe it's a naive question. So birds actively incorporate plastic in their nests.
Yes.
Just saw a presentation where on plastic we find a community of invertebrates. Do you think that there's a possibility that seals actively select plastic to eat as a dish of food source?
Well, I think it could be possible, yeah, because I know that there are a lot of studies on like copepods and fish that they actually ingest it because it looks similar to their own food. I don't know about seals because they mainly eat bigger, like fish, for example. But I think, yeah, it could be possible because they like it. Maybe they see it as a toy or they, yeah. I think also for the captive seals, maybe they have, I'm not sure, but maybe have some plastic toys inside their aquarium, let's say, and then they also, I don't know, get pieces inside. The thing is that we only found microplastics, so no bigger parts, which maybe shows that they didn't actually select it because they were all super small.
That brings me to my second. This topic is new for me, but so you say you only find microplastics. Is there a possibility they eat plastic? Plastic gets degraded, and then it forms microplastics, and so they strengthen the plastic problem.
Yeah.
Is this the case, or is it like no? It's only microplastics.
I think it's for sure possible because the stomach and all the acids inside will also break down plastics, and if they are already staying in the environment for several years, I'm sure they're fragile and they break down, maybe even faster because they are ingested. I would say yes, because also there was one piece which was a bit bigger than all the others, but still microplastics, and that one was very fragile. Like I tried to take it, to put it under the spectrum, but yeah, it broke. So yeah, I think yes. And also because they have a quite big range where they live, they can also transfer, yeah, when they ingest it in, for example, Newport in Belgium, and they fish somewhere else, then they can also transfer their plastic.
And one more question, why birds and seals? Why not birds, different species of birds, or seals and other invertebrates? Why these two groups?
Well, the planning in the beginning was to actually investigate both of the species of them, but because of the avian influenza, it was discouraged to do. So that's why we came up with something new.
But still, why birds and seals? Why these two?
Well,
what is the link?
so the project is based in Newport, which is also known as a typical haul-out site for seals. So they rest there after foraging. So most of the time they actually come in the afternoon to chill in the harbor to digest their fish. And it's a very big bird reserve, and there are a lot of species. So that's why we actually combine them, because they are living quite close by. The problem was then, because we were only allowed to go in the bird reserve for one day during the cleanup, we didn't get time to investigate other species because they were not breeding yet, or they're not brown breeding species. So yeah, we actually had to adapt the plan a bit. And for me, it was also a bit hard to actually keep the two of them because, yeah, Canadian goose are also an exotic species, and then the seals were not only from the Newport area. Yeah.
Yeah, thanks for your presentation. It was very good. I really like that you did your validation with spike samples, and I really like that you used your JList as well for your validation. So, well done. I just wanted to ask you, so when you were collecting these thesis, I understand that you also consider the wild seals were the ones that were in rehab. Is that the case? So when you were collecting, what is the protocol for collection? So how long have they been in captivity before their feces were collected? And like, I mean, I don't know how many times they go to toilet a day. So how many times they excrete a day? So like, how was your sample collection in that sense?
So, yeah, we always asked beforehand, then we would clean several containers and we would bring them to the sanctuary so they were ready to be used when there was a seal pup. So then they went to collect the seal to take it into rehabilitation. And then they tried to take the first species after they arrived. For example, for one of them, it was super little, so they actually also collected the second one, but it was still super close by the time that they got rescued. Some of them, yeah, I mean, most of them are sick, so some of them also contained like parasites, and yeah, they had diarrhea. I don't know if you have a good word for this.
That's a good word.
Okay. Which made it a bit hard to collect it. But yeah, it was as soon as possible. So the first species that they had.
And then you were showing a photo there of one of your samples that looks like little cracks, and you say that it was because of the nature of like your samples increases that was difficult. So I was just wondering if you think that your digestion process perhaps didn't work really well, that it would have that applying more.
My methodology was actually quite a big part of my thesis because I needed to wait for the seal samples to arrive. So in the beginning, I mainly did methodology development. So I tried several steps to include. So for example, the normal methodology was without extra sieving steps. I did mention it because of the time, but in between the digestion steps and the filtration, there were sieving steps to get rid of smaller debris, because I have actually a picture. Mm-hmm. Yeah, so all the samples looked like this, which is a lot. And then when you put a sieving step with a sieve of 50 micrometers, which was also my lower limit now, it was way better, and I was also able to filter, because still I needed to separate some of the debris on several filters, because it was too much for the filter to do it, and then it would just stay on top. So yeah, it's the additional sieving step, and I also tried to include a bleach step. So I did half of the samples in that batch, I did it with bleach, and the half of them without, and I felt like it was not a big difference. So it didn't make a big difference in between the two, so I actually left out the bleach step again.
That affected the colors as well?
Normally not. It was tested already in several studies, and they said it was not. But yeah, of course you're never sure, because I'm sure that the bleach also gives the colors.
I have one last question. Okay, so just now into your birds, why did you have such a small bird sample?
Yeah, it was because there were no more bird nests at the time.
Okay.
Yeah, that's the only reason actually.
And how far, when you were taking photos, how far from the nest?
It was 1 meter, like from up. But it was this was possible because it is an invasive species, and it's not super bad if you disturb them. I think for actual protected species, you should.
Are they invasive or migratory?
Invasive or exotic. That's what people told me.
Okay, so they are Canadian geese. yeah?
Yes.
Just going for a nest there.
Yeah, they're known to actually eat everything from the estuary riverbanks, and they destroy a lot of plants, which is super bad for the other birds in the environment because they feed on the associated invertebrates inside the riverbanks. So they're actually very harmful for the other bird species, and yeah.
So last question, I think, I promise. Two things. Well, yeah. So in terms of the distance to the bird's nest, do you think? I mean, if you're like I say, like invasive species, if you were like dealing with the species that you would be protecting, do you think that's a good distance to keep from a bird's nest?
I would say no, because a lot of birds are also on top of the nests, so you would not be able to see anything. A lot of studies which use photographic for nests do it after the breeding season, which is probably a better option, or like...
That was maybe my next question. When
yeah.
is a good time to do that?
I don't know, birds have different seasons, I guess it would be kind of now.
No, not specifically. So obviously if you're going for a certain species, you would know when they start breeding and finish breeding. So yeah, what I mean is that you go at the end of the season.
Yeah, right.
So you're not disturbing.
Yeah, yeah, true.
They can just abandon their nest and
Yeah.
disturb it. So we won't use way
True.
too close to the bird's nest.
Yeah, I know that in one study that they didn't, say that this was a good methodology because nests degrade quite easily or like quite fast. But I think it also depends on where the nest was and what are the environmental.
What you're looking for is not
Yes.
to really consider. Okay, thanks very much.
Thank you very much, Ellie.
Question. I saw something, a minor mistake in your presentation. Is it PowerPoint or is it PDF?
Yeah. PowerPoint.
Oh yeah, PowerPoint. Can I change something quickly? Never mind. Can I change it? No, never mind. It's okay.
All right, so closing this session and this symposium presentation in this room is Lucas, right? Lucas, you have 15 minutes. 15 minutes to present this release.
Thank you very much. Welcome everybody to my presentation about anthropogenic disturbance and behavioral ecology of green sea turtles in a Brazilian marine protected area. So first of all, balancing conservation and ecotourism. Currently, marine ecotourism has seen an unprecedented growth in the last couple of decennia and is known to be one of the fastest growing tourism sectors globally. It is not only often praised for its increase in awareness and increase in wildlife education, but can also benefit the well-being of humans itself. However, it is not always as sustainable as on paper. Often with the increase in pristine habitats, humans can have effects on wildlife behavior. So there are also a lot of conservation concerns in this growth. For instance, behavioral disruptions by human presence, but also altered feeding patterns subsequently, and also stress responses, just to name a few. We currently have a limited understanding of how ecotourism affects wildlife, especially in pristine, unhabituated populations, and there's a rising need for site and species-specific, science-based conservation management. Therefore, we were set out to study Colonia midas, also the green sea turtle, and you can see a beautiful image here I took myself in Brazil on the screen. It is not only a globally endangered species, but it's also a charismatic flagship species, meaning that in tourist destinations or ecotourist destinations, it is often one of the biggest attractions. Therefore, it is also often exposed to very high tourist exposure. Alongside this, it's also called an umbrella species or categorized an umbrella species, meaning that by conserving this particular species, you often indirectly conserve a multitude of lower trophical levels and habitats as well. The neotropical life cycle of this species begins or starts at the northern Brazilian coast where they hatch, after which they gradually disperse along the southern Brazilian coastline. Here, they inhabit ontogenetic stenotrophic site fidelity, meaning genetically in different stages of their life they inhabit different habitats. Stenotropic is that they are very particular or specific in the environmental conditions this habitat has, and site fidelity is that they stay really in this habitat. I will later come back to why this is very specific and important. So first, a couple of human-turtle interactions we know about. Anthropogenic disturbances like approaches, which can cause flight initiation, physical harassment, which can cause feeding cessation, boat noises, which can reduce resting periods or reduce feeding periods, tourist exposure, which can increase vigilance. Also, once again, these are just a few of a multitude of anthropogenic disturbances. All of these anthropogenic disturbances can cause long-term ecological consequences, many of which still unknown in turtles, green sea turtles. Most studies currently have been done on phototaxis of hatchlings, with very limited studies on behavioral ecology and especially circadian rhythms, with only 3 out of 286 on circadian rhythm. Therefore, we identified a key gap in knowledge with our limited understanding of how tourist exposure affects sea turtle behavior, especially circadian rhythm and habituation. Our objective was to evaluate how anthropogenic disturbance affects circadian rhythm and also escape behavior in sea meadows. We did this by doing 2 parallel approaches. One was focal behavioral observations to assess circadian rhythm, and the other one was human approach trials to assess habituation processes. We compared 2 sites, the high tourist exposure site, which historically has seen very high abundances of tourism, and a low tourist exposure site, historically very minimal tourism exposure. From now on, I will trust them as high and low for simplicity. Our hypotheses are that the low side has increased flight initiation distance due to unhabituationness to human presence and a prolonged resting period due to less human disturbance early in the morning by tourism, and also that the high side has reduced flight initiation distance due to habituation and phase shifting resting periods due to human disturbance. As I told you about the site's fidelity of the species, and they migrate south towards juvenile foraging habitats, Areal de Cabo, just a bit north of Rio de Janeiro, is such a very critical forage habitat where young turtles come to develop before sexual maturity. Our high site, as seen here in red, is alongside a cove, called also the Graçanha Cove, and is accessible by land trail and therefore has seen historically very high abundance of tourist exposure. The low side has no accessibility or tourist infrastructure, and therefore it's more difficult to come, and maybe you see an occasional spearfisher, but very low tourist pressure. The fact that they're so close to each other, I will come back to that later. So first of all, our focal behavioral observations. We sampled 64 turtles, averaging or totaling 790 minutes in total. We did in-water observations ranging from 3 until 15 minutes. We did this from a non-intrusive distance previously documented, and we scanned both the sand reef interface and the coastline to prevent any behavioral state bias. Our analysis, we adopted an ethogram from previous marine reptile studies. However, the ethogram was big. We only looked at solitary behaviors because the inter and intra-specific behaviors were very limited. We had 4 time blocks in between 8 and 2 o'clock, all 1.5 hours, and we documented the relative time feeding, resting, and swimming. We did permanova to look at circadian rhythm differences between the 2 sites, and we did Wilcox per time block per behavioral state to look at specific behaviors. The diver approaches consisted of 47 in total diver approaches. We had a 2 standardized diver approach methodology also adapted, where we stayed both at a non-intrusive distance. One would film the approach and the other one, in this case me here, would swim towards the turtle. I would never touch them. And we did this for 2 states, both feeding and resting turtles, to assess state-dependent habituation. We manually post hoc did flight initiation distance estimation with the help of a reference difference, which was a half meter of my arm. We cross-validated this for security between multiple students and professors, and it's biased, can be biased. Increments of a half meter, and alongside this we also did reaction severity scale. We adopted this as well, with including startle, displaced, and tolerant. We did Wilcox for the FID, and we did chi-square for the reaction severity analysis. The photo ID for site fidelity control, we leveraged i3S Planet software with used facial scale recognition to identify each turtle individually. This avoids double observations of the same individual per day, which was vital for the flight in chaseness distance protocol, and it had a match accuracy of over 95% previously documented. This ensured data independence between turtles, but most importantly, it ensured that the two sites that were very geographically close were actually two distinct populations, and this was the case. So what we found was altered activity patterns between low and high. We saw, for instance, that the high side here, when looked at feeding, after their resting period went down, high is orange by the way, they started to feed, significantly higher than the low side. The low side had complete suppression of feeding throughout the day, but I will come back to that later as well. What we did see was that the low side indeed had more resting until later on in the day, while the high side had lower resting. And the low side, after their resting period was over, at 12:30, they started swimming immensely. We come back to that later as well. The FID and reaction severity showed us also that the less habituated turtles at the low side showed significant increase in FID compared to the high side. And also when looked at the reaction severity, we saw that feeding turtles had significantly more startle responses than their counterparts on the high side, meaning that there is a distinct habituation process happening. When looking at resting turtles, we see that there's no difference or significant difference in between high and low side, and also with reaction severity, there was no significant difference by chi-square test. So back to the complete suppression of feeding at the low side. What is the behavioral mechanism? So we saw a very heightened vigilance in unhabituated populations, where if we approached or observed the turtles at the low side, even though they were busy with feeding, they were often seen that after the approacher was present, they would swim towards the open water and actively circle around us in a state of vigilance. This is possibly because they perceive humans as possible threats, and it caused or triggered an anti-predator response activation. This is also why we saw them swimming a lot after resting. When also looking at the FID and reaction severity, we saw that resting turtles do not significantly differ in reaction. So we saw that feeding was not very much affected by our presence, resting, but only feeding. The energetic consequences are, of course, what we saw, reduced foraging time, but also potential impacts of growth rates eventually, and possible effects on reproductive fitness. What we saw overall was with Paramanova that tourist exposure affects circadian rhythms. We saw different patterns between high and low. We saw site-specific habituation when looking at the FID and reaction severity. And we also saw behavioral dependent response modulation, where turtles inhibited different behaviors, responded differently on human approaches or impulses, in lining with the optimal vigilance theory. Regarding the circadian rhythm, everything combined, a theory that is most in line with my current data is something called the anthropogenic adaptation continuum, where our sites represent different stages of wildlife adaptation to humans, and they come from what they call a naive population through a process of xenotropy to a more habituated situation. Our low side was unhabituated, our high side habituated. For conservation implications, this means a one-size-fits-all model is ineffective, meaning that you really have to look specifically at certain historical components of each site for site-specific management. This can be, for instance, temporal restrictions into certain resting periods on the day, but also an exposure-specific approach distance for sites that have historically low exposure that are naive. So maybe you should stay at a bigger distance than sites that are habituated to humans. With the photo ID, we also saw that small differences matter, small distances. So undisturbed refugee zones can be very beneficial for marine protected areas, for instance. Our current limitations are, of course, the observer effect, as almost in all behavioral studies, with in-field observations. We have seasonal variation. We only did it in March and April. The whole year round would be more beneficial to see if there's maybe seasonal variation. And individual tracking with what we saw with the cameras on the back, that would have been very beneficial if we can do that more often also in Cabo. So long-term or future research directions can look at the long-term monitoring of individuals. It can look at physiological studies, for instance, measure stress hormones, which is very often seen to be an effect of affected circadian rhythm or changing in circadian rhythm. And population impact, so assess growth rates in between populations, in between naive and habituated populations. The conclusion is that chronic tourist exposure significantly modifies green sea turtle behavioral ecology, with both adaptive and potential harmful consequences. Behavioral plasticity does exist, it does occur, however, habituation takes time, and we don't in particularly know how long that habituation process takes. And the hidden cost in between this process is still unknown, because we only saw two extremes, so to say. Therefore, we can recommend site-specific management to be crucial for the conservation of a lot of populations. And I believe that there is a balance, that the balance is possible with science-based guidelines. I would like to thank both my professors and Luciana for her expertise in the field of photo ID and individual tracking, Brunio, and of course the EMBHC program. Thank you very much.
Thank you, Lucas, 14 minutes and 30 seconds, so very nice and useful. Thank you very much.
That's good, yes. So I'm a seagrass researcher, and I know these green turtles, they love seagrass.
Yeah.
Is there no video or any picture I saw in seagrass there? So what is the situation there?
Sorry?
What is the situation there?
In both sides.
Yeah. Is there seagrass with female?
Well, it's actually so both sides are very similar to each other. They both have rocky shores and then eventually a sand bed. They both have abundance in both green, red, and brown algae. However, what we saw is that on the right side, so on this side, there is a bit more ulva than on this side, which they tend to prefer. But on this side, they also eat a lot of red algae. And I looked more closely into this because I wanted to know the process behind it. And there's actually a lot of studies coming up now. I think they're a couple of years old, that for a turtle, it does not necessarily, they don't really like a particular macroalgae species. They're very much driven by the microfilm that is on it. So even though they eat ulva a lot here, this study, or the two studies that I wrote about microfilm around something, that is more important to why they eat something. So the microfilm around ulva here is something they tend to like, while this side has more red algae.
Is it mother algae or seagrass?
Not little seagrass, little.
Because there's not a lot of seagrass studies in Brazil, South America.
You saw like occurrence of seagrass, but I didn't see them eating. They were very much on rock feeding. Also, the adapted ethogram that we used, they had all kinds of positions where they ate from jellyfish. It was a very big, with also durations, amount of bites, and then amount of breaths and everything. But we really only looked at durations. But I also, for future research so that they can use my data, I did write down what they ate specifically, when they ate it, how many bites. So it is all there in the data. But
another question, but so you have ecotourism, people want to see turtles, a lot of money goes into habitat restoration or can go. And now we have a study that says maybe we should leave them for several hours a day. How do you balance this? Because people want to see turtles, and if you say, okay, half a day we don't see turtles, you don't get the money for habitat restoration.
Well, actually,
how do you see this balance? How should we?
Well, in my current experience, it's already been widely used in the field. That, for instance, there are some boat tours in Ariel de Cabo, but none of them are, or they are all prohibited from this particular bay. But they do go around quite a lot. And it is already implemented that certain bays have certain temporal restrictions with time. And I also saw this on holiday a lot. I'm a diver myself and a snorkeler. And it's very often seen that you can just say, okay, from 2 until 3 o'clock, you with a boat tour or with tourists are allowed to be here. But after that, you have to be out of the bay.
Maybe shift locations.
Yeah, shift locations. And you can, for instance, say, okay, this site has already been habituated, so you can go there first, and then to the unhabituated site, you need to go later on the day. No worries.
Hi, Chris. Thanks very much for your presentation.
No worries.
Very interesting. Okay, so you said you had, was it 64 turtles in total?
Yeah.
And how many of them were photo ID?
All of them.
All of them?
Yeah. It was actually the girl I spoke about last, the Luciana. She was also, I was in Ariel with the whole turtle team. We lived together and did everything together. And we did, we all studied turtles with all a different topic. And we did all the photo IDs together, and she also helped me with the observations. But yeah, she basically, after the video, so after the 3 or 15 minutes, the approach was that you would go towards the turtle and scan both facial skewed. So first you stayed, of course, as long as possible to the non-intrusive distance. And then afterwards, when you're reaching the 15-minute mark, you would go and come.
That's excellent for future work.
Yeah.
To actually have that.
Exactly.
And then one thing I'm missing here is, so this all happened in Crimea, right? So
Yeah.
Ukraine and the Tanzania.
Yeah.
Okay, so how distant are these two sites?
I think 1,000 meters only.
So how many meters?
1,000 meters only.
1,000 meters.
Yeah.
Okay. And then, so if you photo ID that, and you also mentioned there was very little mixing between the populations, how much mixing is there? So how many turtles did you find in one place that you also saw?
So in total, we found 2 turtles at the low side. Of the 64 or 67 was kind of equally divided across sides. We found 2 turtles of the right side on the left side.
And so, I'm going to go to questions. For two more things. So, how do we differentiate, so behavior studies are very difficult, so I completely sympathize with that. So how do we differentiate when we're saying they're swimming because they're stressed or they're resting because they're stressed? So how do we actually differentiate that?
So that is indeed very, very difficult. What was a remark in our ethogram again is the way they swim. We also had observing us, so the times that at the low side they would swim around us and have a constant eye on us, it was very typical behavior for the low side to really go towards the water column and observe us, and we did write that down. But eventually we just wanted to see the big kind of differences in the 3 biggest behaviors. But to really say that if they're stressed or not stressed, that's very difficult to say. That's why we wanted to employ the flight initiation distance and the reaction severity to see how the turtles differently react to us. Because with only the circadian or the behavioral observation data, that might have been insufficient to address those questions.
So you talk a lot about the circadian rhythm, and I think there's more important stuff in your thesis than that.
Yeah.
But what's the definition of the circadian rhythm?
It is their activity pattern throughout the day. And you can see that with, for instance, sleep rest, but it goes beyond sleep rest. So it also goes, for instance, awakening, then feeding, then doing something else and then feeding again. It's really there.
But what's that influence by?
It can be anthropogenic disturbance. It
No,
can,
what is the main thing? So let's think about the circadian rhythm for us. What does it mean if you say, oh, my circadian rhythm is off? What does that mean?
that your natural cycle is affected.
Yeah, but what is that affected by?
Any external cues, it can. I don't
Main thing? Light? Maybe?
Yeah, light, but yeah, light.
Yeah.
But we don't introduce a lot of light into their natural habitat.
Yeah, so that's why when you're saying that they are having their circadian rhythm affected, it's a bit of a strange thing to say that because they're changing the activities that they are behaving in a certain way in certain times,
Mm-hmm.
but it doesn't mean that they're changing their circadian rhythm all of a sudden. Do you know what I mean?
I do understand what you say, but what I did find was that their sleeping pattern is affected in the different two sides.
Okay.
So I think there's more studies
So
to be done.
it's steep for you.
Not only, but it is one of the components of.
Thanks very much.
Thank you.
Right? Speculation, Lucas.
Thank you.
Oh, sorry.
Oh.
I forgot. Christina. No, we do have time, but I thought I went through the 2 minutes. Sorry, Christina. Please.
About this last issue, about resting. What do you mean about resting?
So resting is
Or just
so for turtles it's very difficult to assess their resting period because they rest with their eyes open. But it's mainly if they go under a rock and kind of I don't have a video of this, but they're often small caves and they put themselves below so that they're not affected by the waves and the currents constantly, so they stay stationary and they do nothing there. So there was also a distinction which you had to pay attention to is in a previous presentation you saw this actually, that they also go under the rocks to scratch their shell to remove barnacles of kinds and everything. So you did need to that was also a category in the epigram, but that was not resting, not considered resting.
is resting affected by the disturbance, the disturbance. Is affected.
Well, we saw that the high side, so the one that saw a lot of tourism, especially after around 10 o'clock, they abandoned resting earlier than the low side, and which might be because of their historical high tourist influence, and tourism does go paired with a lot of points.
Is there any difference between among species of different age?
Sorry?
Is there any difference between different age classes?
That's a very good question. So about their life cycle, what we found in Areal de Cabo is mainly juvenile sea turtles, but there are a few more adult ones. And to prevent this bias, we excluded them from our analysis. So we tended to look at only smaller sized individuals, because we also saw, for instance, that bigger individuals are slower, more habituated. So that would not really make any effect on them. Stuff. But the main population in Areal de Cabo is juvenile. The older, more mature ones, they stay more along the northern coast, and they have site fidelity, inhibit site fidelity there.
Anybody has another question? No? This time for real. Congratulations, Lucas.
Thank you very much.
Brings us to the end of this presentation part of the symposium. Congratulations to all of you. I think now we all have lunch here, and then you go back to the SM and see each other tomorrow. At 2 o'clock, yes, you go in class.
then system analytics, you'll do some research, which you can buy yourselves. And one will be there with some explanation about eco and this.